जगत का कल्याण कर माँ, जगत का कल्याण कर माँ, तुम हो विघ्न विनाशिनी। जयति जय जय माँ सरस्वती, जयति जय जय माँ सरस्वती, जयति जय जय माँ सरस्वती।
दीप ज्योति परब्रह्म, दीप ज्योति जनार्दन, दीपो हरतु मे पापम, दीप ज्योतिर्नमोस्तुते। शुभं करोति कल्याणं, आरोग्यं सुख संपदां, दुष्ट बुद्धि विनाशाय, दीप ज्योति नमोस्तुते। I take this liberty to invite the dignitaries with a request to come on to occupy the distinguished chair on the dais. To maintain the decorum of the program, I humbly request all of you to keep your mobile phone on silent mode. Let us now take a moment to extend our warm greetings to the distinguished guest seated on the dais. For this, I would like to request Professor Harit Jha sir to kindly welcome our Honorable Vice Chancellor, Professor Alok Kumar Chakraval sir, by presenting him with a plant sapling. I would like to request Dr. Ashish sir to kindly welcome our guest of honor, Professor Min Fong Lin sir, by presenting him with a plant sapling. I would like to request Professor Chandrasekhar Gahan sir to kindly welcome our registrar, Professor A.S. Randive sir, by presenting him with a plant sapling. I would like to request Dr. D.K. Parihar sir to kindly welcome our Dean, Professor Rajendra Mehta sir, by presenting him with a plant sapling. I would like to request Dr. Sanjeev Kumar sir to kindly welcome our head of the department, Professor Harit Jha sir, by presenting him with a plant sapling. I would like to request Professor Renu Bhatt ma'am to kindly welcome course coordinator Dr. Santosh Kumar sir by presenting him up with a plant sapling. I would like to request Mrs. Alkaika ma'am to kindly welcome Mrs. Ten Fen Lin by presenting her with a plant sapling. सभी का बहुत बहुत धन्यवाद. A single phosphate can turn a protein straight. That is the power of phosphorylation. With this note, I cordially invite Professor Rajendra Mehta sir, Dean, School of Studies of Interdisciplinary Education and Research, Ghasidas, Vishwavidyalaya, Bilaspur, for welcome address.
परम आदरणीय कुलपति महोदय, गुरु घासीदास विश्वविद्यालय, आदरणीय रजिस्ट्रार साहब, गुरु घासीदास विश्वविद्यालय, आदरणीय प्रोफेसर मिंग फोंग लिंग, आदरणीय प्रोफेसर संतोष कुमार, प्रोफेसर हरीश झा, मेरे सामने बैठे हुए मेरे शिक्षक साथियों और पार्टिसिपेंट्स, सबसे पहले मैं अपने प्रेरणा स्रोत हमारे वाइस चांसलर प्रोफेसर आलोक कुमार चक्रवाल साहब का तहे दिल से स्वागत करता हूं। उनकी प्रेरणा से ही आज हम इस कार्यक्रम को करने में सक्षम हो पाए। मैं स्वागत करता हूं प्रोफेसर मिंग फोंग लिन का, जो कि अमेरिका से आए हैं। अभी बातचीत में उन्होंने बताया कि काफ़ी सारी फ्लाइट्स को उन्होंने बदल करके बड़े कठिनाई से वो यहाँ तक पहुँचे हैं। आपका स्वागत है सर। मैं स्वागत करता हूं हमारे कुल सचिव महोदय प्रोफेसर A.S. रणदिवे जी का, जिनके पास हमारे लिए वक्त है, जो हमारे कार्य को करते हैं बिना रुकावट उसका मैं हृदय से स्वागत करता हूं। मैं स्वागत करता हूं बायोटेक्नोलॉजी विभाग के विभागाध्यक्ष प्रोफेसर हरि झा और उनकी एंटायर टीम का, जो इस प्रोग्राम में किसी न किसी प्रकार से अपना सहयोग प्रदान कर रही है। मैं वेलकम करता हूं, स्वागत करता हूं ज्ञान के कोऑर्डिनेटर प्रोफेसर राव साहब का, और आप सभी पार्टिसिपेंट्स का भी हृदय से स्वागत करता हूं जिन्होंने इस ज्ञान के प्रोग्राम में अपनी रुचि दिखाई है। थैंक यू वेरी मच।
Thank you very much, sir, for your warm and inspiring welcome address. From oxidation to restoration, redox is the silent regulator of life. With this thought, I warmly invite Dr. Santosh Kumar, sir, Associate Professor in the Department of Biotechnology and Coordinator of this course, to kindly share his insights and throw some light on the course.
Yeah, good morning everyone. Honorable Vice Chancellor, Sir Professor Kumar, Esteemed Registrar, Professor K.S. Bandare, our distinguished Chief Guest, Professor Ming Pong Lin, from University of Nebraska Medical Center, Omaha, Nebraska, USA, Professor R. Mehta, Dean of School of Study of Interdisciplinary Education and Research, Local Coordinator GIAN, Professor M.C. Rao, colleagues, aspirant and participant, good morning to all. It is my privilege to present a brief overview of our GIAN courses on multi-omics crosstalk on the phosphorylation, redox and endocrinology for therapeutics, being organized by the Department of Biotechnology, Guru Ghasidas Vishwavidyalaya, from 27 to 31st October 2025. This course provides participants with the advanced understanding of how proteins, the key function molecules, regulate diverse cellular activities through specific modifications. It explored the cross-regulation among protein, steroid, and free radicals at the post-translational level, highlighting their role in the gene expression, physiology, and disease. Comprising total 14 lectures, the courses focus on post-translational regulation of protein function to explain how molecules, signaling molecules, influence health and how their dysregulation lead to the disease. Understanding these pathways support the development of targeted and specific therapeutics strategies. Professor Ming Hong Lin, Professor of UNMC, USA, will highlight about protein phosphorylation and other post-translational modification, redox regulation involving oxidant, antioxidant, and signaling pathway, as well as hormonal regulation, cell cycle control, and their role in the cancer mechanism. Dr. Rupesh Kumar Srivastav from AIMS Delhi will focus on the cancer signaling and targeted therapeutics, explain crosstalk regulation in carcinogenesis and guide data analysis during practical training experiment. Dr. Santosh Kumar Balot from NCCS Pune will enlighten about various types of post-translational modification in cancer, such as the phosphorylation, glycosylation, and ubiquitination, and explain how these modifications can be targeted for therapeutic intervention. Overall, these courses has been designed to integrate theoretical knowledge and with practical understanding, offering participants an opportunity to engage, discuss, and deepen their comprehension of cellular signaling, regulation, and therapeutics targeting. Our ultimate goal is that by the end of this program, every participant leaves with the clear understanding of the epigenomics, its application, and their future aspects, and with the confidence to integrate their knowledge in their own research and academic pursuit. I would like to thank GIAN for enabling us to bring this program to our university, and extend my gratitude to all resource persons for sharing their expertise. I also appreciate the effort of my colleagues, organizing committee member, and our enthusiastic participants, without whom this course would not be possible, has been possible. With that, I warm welcome all of you to this intellectual journey into the fascinating world of the epigenomics. Let us make their 10 days, then 5 days truly enriching the collaboration experience. I'm hopeful that by the end of these 5 days GIAN Pro courses, each participant will not only gain the advanced insight into the molecular mechanism of the cancer, but also develop the broader perspective on how these findings can be translated into the protein treatments. With that, I warmly welcome all of you to these 5 days short-term GIAN course. Thank you.
Thank you very much, sir, for providing us with an overview of the course. It is not just what proteins are made of, but how they are modified that defines life's diversity. With this thought, I humbly invite Professor A.S. Randeve, sir, Registrar of Guru Ghasidas Vishwavidyalaya, Bilaspur, to kindly address the gathering on this special occasion of inauguration of the GIAN course on multiomics, crosstalk of phosphorylation, redox, and endocrinology for therapeutics.
Thank you. Respected Vice Chancellor of our university, which is a university with difference, we proclaim. Under his leadership, the university has developed itself into a different level. So he is with us. Main resource for this GIAN series, Professor Ming Fong Lin from University of Nebraska. Every university over there in USA has its own medical center. So he is from that medical center of University of Nebraska. Dr. Fen Lin, who is life partner to Professor Ming Fong Lin. Professor Mehta, Dean of the school. Professor Jha, Harit, Head Department of Biotechnology, the course coordinator, Dr. Santosh, sitting in front of us with distinct academicians, and GIAN coordinator for Guru Ghasidas University, Professor Rao. Dear students, such type of initiatives, which are termed as global initiative for academic network, will definitely provide us with an opportunity to interact with experts in different areas, and that will not be a lecture just telling something on the surfaces, because this is 5-day rigorous training program along with the laboratory sessions also. So such type of event will give you a different perspective to your research, your personality, your way of thinking, and taking up the challenges. So I wish you every success in this endeavor. I will like to congratulate Department of Biotechnology for arranging second such program in this academic session, and total fifth, Professor Rao, I suppose this is fifth of this academic session. This reflects the dynamism of the entire university under the leadership of Vice Chancellor Professor Alok Chakravarti, sir. So I will like to take this opportunity to thank him in a very different way from the core of my heart, and from the side of all participants in this in an entire university academic fraternity, for making these events so quickly in succession in every year. Thank you, sir. Thank you, everyone.
Thank you very much, sir, for your valuable and inspiring address. Your words have truly enriched the spirit of this inaugural session and set the tone for the workshop. From molecules to moods, endocrinology connects chemistry to consciousness. With this thought, we now move ahead to the next part of our inauguration program. It gives me immense pleasure to introduce our esteemed resource person, Professor Ming Fong Lin, from the Department of Biochemistry and Molecular Biology, College of Medicine, University of Nebraska Medical Center, Omaha, USA. Professor Lin is a globally recognized scientist and educator whose research has made remarkable contribution to the understanding of prostate cancer biology and molecular mechanism of cancer progression. He completed his BS degree from School of Pharmacy at Kaohsiung Medical College in Taiwan, followed by MS degree from Institute of Molecular Biology at National Tsinghua University, Taiwan. Later, he pursued his PhD at Roosevelt Park Memorial Institute Graduate Division, State University of New York at Buffalo, United States of America, where his thesis was focused on biochemical characterization of prostate-specific antigen and prostatic acid phosphatase, two key biomarkers of prostate cancer. After completing his doctoral studies, he carried out postdoctoral research at the Louisiana State University Medical Center, New Orleans, USA, before embarking on a distinguished academic career. Professor Lin has served in various academic and leadership positions, first at the University of Southern California School of Medicine, and later at the University of Nebraska Medical Center, where he has held several prominent roles, including Vice Chair for Research, Vice Chair for Academic Affairs and Faculty Development, and Professor of Surgery and Urology. He also holds courtesy appointments at the Epley Institute for Cancer Research and the Department of Surgery at University of Nebraska Medical Center. Over the years, Professor Lin has been the principal investigator or co-investigator on numerous prestigious research grants funded by leading US agencies such as the National Cancer Institute, National Institutes of Health, the American Cancer Society, the Department of Defense Prostate Cancer Research Program, and the Cap Cure Foundation. He also holds several US patents related to prostate cancer diagnostics and therapeutics, including the establishment of novel prostate cancer cell line models that have advanced the field of cancer biology. His outstanding research accomplishments have earned him numerous honors and awards, such as the UNMC Distinguished Scientist Award, the Research Innovation Award, and the Distinguished Alumni Award from Kaohsiung Medical University, Taiwan. Professor Lin is also a fellow and active member of many renowned international scientific societies, including the American Association for Cancer Research, the American Society for Biochemistry and Molecular Biology, the American Society for Cell Biology, and the Endocrine Society. In addition to his outstanding academic and research contributions, Professor Lin has rendered exemplary committee and community services at the University of Nebraska Medical Center. He has actively served on numerous institutional committees, including faculty grant review panels, graduate and medical student research committees, and academic affairs and development boards, often in leadership roles such as chair, vice chair, or coordinator. Professor Lin's commitment to scientific advancement is further reflected in his role as reviewer for numerous prestigious international journals, including Cancer Research, Clinical Cancer Research, Carcinogenesis, Oncogene, British Journal of Cancer, Molecular Cellular Endocrinology, Pharmacological Reviews, PLOS One, and the Proceedings of the National Academy of Science, USA, among many others. He also served on the editorial board of reputed journals such as Clinical Medicine Urology, Biomed Research International, Cell and Bioscience, the Kaohsiung Journal of Medical Sciences, the Scientific World Journal, and the Open Enzyme Inhibition Journal. Through these roles, Professor Lin continues to uphold the highest standard of scientific integrity, peer review, and mentorship, contributing immensely to the global biomedical research community. Professor Lin has published more than 135 research articles and book chapters in reputed international journals and has delivered invited talks at scientific meetings and conferences across the United States, China, Taiwan, Singapore, Italy, and Germany. Alongside his research, Professor Lin is deeply dedicated to teaching and mentoring. He regularly teaches biochemistry, endocrinology, cell and cancer biology to graduate, medical, and pharmacy students, and mentors numerous undergraduate, PhD, and MS graduates, and postdoctoral scholars. He also supports several training programs of minority and international students in biomedical research. In recognition of his lifelong contribution to science, education, and cancer research, Professor Lin continues to inspire students and scientists worldwide. Ladies and gentlemen, please join me in warmly welcoming Professor Ming-Fong Lin, a distinguished scientist, educator, and visionary researcher whose work continues to make a lasting impact in the field of molecular oncology and medical biochemistry. With great joy, I invite Professor Ming-Fong Lin to kindly grace the podium and deliver his words as guest of honor for this GIAN course. Over to you.
The first Honorable Vice Chancellor and respected the registrar, the dean, and the chairman of the department, and all the faculty members, the students, everyone. Good morning. It's my great honor, privilege to be here as a guest honor for the lecture for our multiomic course. Okay. We know the functional molecule in the cell is protein, Yeah, so RNA, DNA, yes, they are very important. I cannot ignore it, but the really functional one is really the protein. But the protein cannot function alone. They got to be modified. That they can be so they can do their respective job. So they can be regulated by different kind of modification. And that's what is the topic we are going to discuss in the following 1 week. Focus on the phosphorylation. And not only phosphorylation, actually it interacts crosstalk with the redox. So we always think the redox may not be so important. Be honest with you, when I was a student, I consider the free radical could be a mimic. It's not really an important one. But in reality, now we know the free radical not only involved in our aging, also involved in essentially all the disease can be involved. The cancer, the heart disease, the diabetes. So a lot of disease, I mean, it's really, we cannot forget about the free radical. Then steroid hormone, yes, how we are a male or how can we be a female? It's regulated by basically steroid hormone. So if you think the signal pathway is independent each other, it's not, it's wrong. They actually crosstalk, they interact, like in the traffic, we always think one-way street, but it's not the case. They always crosstalk, they always a cross section. So that's what we are going to focus in our discussion in the in this week. So finally, I'd like to thank you for the opportunity for invited by the Professor Santosh Kumar for the opportunity. I also appreciate the support by the GIAN program for the opportunity. And we have been, my wife and I have been here. We really thank you for, we enjoy very much for the hospitality by everyone. Thank you again. Thank you.
Thank you very much, Professor Ming Hong Lin, sir, for your inspiring and thought-provoking address. Your words have not only enlightened us, but also added immense value to the inauguration of this GIAN course. We are truly privileged to have benefited from your insights. We are indeed delighted to share that two more eminent resource persons will be joining us a little later in the program. First, Dr. Santosh Kumar, Scientist D at National Center for Cell Sciences, Pune, a distinguished researcher with deep expertise in cell biology. He obtained his PhD from CSIR Imtech in 2011 and later pursued postdoctoral research at the University of Chicago at Yale University. His pioneering work focused on cellular signaling and protein trafficking relevant to human diseases, particularly dissecting post-trans Golgi network trafficking and characterizing signaling protein complexes formed by the major brain G protein. We are also honored to soon welcome Dr. Rupesh Kumar Srivastava, sir, additional professor at AIIMS, New Delhi, a leading name in the field of cellular and molecular immunology. He is the founder of the Translational Immunology, Osteoimmunology, and Immunophoresis Laboratory. His work delves into the role of regulatory T cells, Th17 cells, and B cells in bone remodeling, paving the way for innovative therapeutic strategies against bone-related diseases. We eagerly look forward to their insightful sessions, which are sure to enrich today's scientific deliberations. Precision therapy begins there, where omics converges, understanding before intervening. We are privileged to have with us our Honorable Vice Chancellor, Professor Alok Kumar Chakravarti, sir, who is a beacon of wisdom and inspiration, and whose dynamic leadership has been instrumental in steering our university toward excellence. Sir, I request you to grace the dais and deliver the presidential address.
Good morning to all of you. Big round of applause for Ming Pong Lin, traveling all the way from Nebraska to this country, and that too in the central part of India, and Bilaspur. and that too along with his wife, because getting support from wife is a big task, and I appreciate you somehow convinced her to travel all the way from Nebraska to Bilaspur. And I hope you will certainly make her, not him, I'm asking about her, to taste all Chhattisgarhi, dishes and kind of local diet and everything, and she'll be happy. A little spicy, maybe you are not happy with that. I've never been to Nebraska, but I hope you are enjoying the climate here. I mean, of course, to the East Coast and other parts of the USA, but not Nebraska I've been to. And I must appreciate the efforts of Santosh Kumar, who has made this possible to make a kind of meeting between the students and the US expert, who is very renowned person, in short of protein engineering, I can say, right? I'm not going to in detailing, but that is the most important thing, which is going to cure the different kind of cancerous diseases. Somewhere I read that probably Russia has developed a vaccine for anti-cancerous disease, and by 2027 we are going to receive that. They are still in the trial process, but protein is the biggest cause of most of fatal incidents these days. So working anything upon protein is really something life-saving, and anti-aging also. Like it can re-engineering and redesigning or modifying protein is also always going to make some sort of good thing to the humankind or the mankind. So my intention, why I'm putting a lot of stress upon protein and all the time protein, but sometimes excess of protein can create trouble for you. That is also one thing, very troublesome thing. So, and what kind of protein you are consuming and what kind of protein you are, whether you are able to digest and consume it or not. But the hangout time is very important. Here will be lecture and on. The problem with us is that we most of the time we focus in the classroom, but your future is hidden in the networking, the hangout time. When there is a tea break, you try to approach Lynn. Sir, how can I be connected with you? You take his email ID. I'm talking to students, I'm not talking to professors, right? Those who are aspirants of, having good work. So, I had been to many good head of the institutions and presidents. They move across the globe, and then smart students get an access to the person, and then what happens, they get a lot of opportunities, maybe postdoc, maybe good research lab work, maybe you are going to work as an assistant, maybe you are going to work as a part of a big medical team, you are going to work upon some sort of medical engineering sort of thing. So there are things on and on and on, and it will happen. I must congratulate Professor MC Rao, and a big hand of applause for him, because he is a person who has been taking care of all this thing. This is consecutive 5th program, and I would appreciate Professor Len if you could, because we are ready to host anybody who is willing to come here and on medical sciences and medical researches, because our students are very talented. Only thing is that they are looking for some sort of exposure and systematic environment where they can go and get in and chip in, and then they start doing something sort of good work. I have all trust among my all students who are sitting here, because they are here, and they are very dedicated students. They can work up to beyond the expectations of world-class researchers. So this is an opportunity. You are going to interact with the people from AIMS Delhi, maybe from somebody who is in yet another expert from US, or maybe number of, and this program is 1 week program, I guess. No, 1 week program. You can try for 2 weeks also, when they can stay longer and they can spend more and more time. And I tell you, important thing is not instruments. Important thing is not gadgets. Important thing is not the machines. The important thing is your brain, your visualization, your conceptualization, your way to resolve and dissolve, your way to evolve how you are thinking about a problem, what is your way of doing the things. You think about 100 years ago when there was no machine, but still people were doing wonderful job. You think about Louis Pasteur. You think about C.V. Raman. You think about Madame Curie. There are many people. That time there was no instrument, but people were working. Today we are slaves of the instruments. We are slaves that okay, this gadget is not there. I can't work because there is no instrument. Think about our own ancient Indian scientists who were doing anything without any kind of instruments. So you just observation is keen observation is the most important. How things are changing. How things are recently. I was which country I'm not, but they started observing the plants leaf, and there was some gold particles in plant leaf. So they started digging it out. They got that there's a lot of gold treasure beneath the earth, and there was some kind of mechanism which was bringing very small particles of gold from the water to the leaves of the plants. So it's all depending upon how you are observing the things and your surroundings, how you're studying things. And I tell you, there is no kind of alternate of your hard work. The more hard work you do, the better result you will get. Intelligence keeps on coming after sometimes you will start learning how things being get done better, and how things are getting really percolated to the place where you want to get it put in and fall in, and things start getting done and materializing things. But the important thing is that how deeply you are motivated to your work. So anybody can have a degree in masters in biotechnology, anybody can have masters in science, masters in medical science and all that. But there are very few, like this year medical award, a medical Nobel Prize, right? So how to avoid cancer, how to survive longer, fasting, how it repairs your. I think it is on fasting only, right? Maybe. So how fasting can keep your body better, and that we had been talking from years and centuries ago, that you have to go for certain period. All beliefs and all faiths and all religions you'll find, but we are scientifically proving that we are getting Nobel Prize. If you are not proving it scientifically, then it's only kind of belief and faith, and keeps on flowing from one place to another. And we say, our ancestors used to do that, and that's what I'm doing here. Not like that. Why ancestors used to do in our Indian traditions and Indian lifestyle, you will find many things are there which can be bring to the world for a better medical treatment, or for a better medication of number of and numerous diseases. So I believe that this is a time for networking. Global Initiative for Academic Network. The name of the program is GIAN, Global Initiative for Academic Network. We are connecting to the rest of the world with this program, and there is a big spell of such kind of events going to happen. It is up to you. You faculty guys, you apply for it. Money is not a problem. Funding is not a problem. How good is your proposal? Evaluators found Lynn's profile up to the mark, and they said, no, we have to invite here Lynn. And I really am thankful that Lynn invited his wife also along. So you also invite other academicians, and they have to come, and we will be really happy to host them. We are having moderate infrastructure. I'm not saying state of the art infrastructure, but we can be up to the market. Good equipments. We are going to invest a lot of money in good research infrastructure, so they can come and have their work here, and you can guide how and better we can get funding from global agencies. There may be a lot of good organizations and entities and agencies who can fund good researches, and you just look into our talents, the students, how they are going to present themselves. You'll find that there are good talents here, and they'll be presenting themselves for a better future and better prospects. So this is an opportunity where you are going to get an insight what is happening in Nebraska, what is happening in entire US and Europe and elsewhere, because he is part of so many journals. He is in part of editorial board, and he is contributing a lot. I mean, I'm sure his index must be very much wonderful, which is going to astonish you, maybe motivate you. So the opportunity will not come again. You don't wait that some other expert will come and then you will again, try to make a kind of interaction. Here you have got an opportunity. You try to interact with him as much as possible, till from morning till evening, because probably he's staying outside the campus, I guess, not on the campus, right? So till he goes to his hotel, don't chase him till his hotel. Otherwise he'll be really afraid of and annoyed also. But he is on the campus. You try to explain and express your desires and wishes, academic wishes, not personal wishes, right? So your ambitions and all that. And I'm sure this is going to be a new beginning in the history of GGV and this department, biotechnology. I'm sure this interaction is going to make a lasting impact upon all students and faculty members, and we are going to have some fruitful interaction which is going to help finally mankind, human being, and the society, and we are going to bring out some solutions like redox. I've been listening, but I don't know what is redox and how it is having kind of impact on oxidation, deoxidation, all that. But okay, fine. I'm not a man of biotechnology, or I'm a man of accounting and finance. So most of the time these things go haywire, and I don't understand much of this. But anyways, predominantly I think most of the time about growth and development of my institution, and this is yet another addition in my developmental effort that this interaction is going to happen. And you all are here. I'm really congratulating Department of Biotechnology, the head of the department, my registrar, who had been all the time, after everything, and he's paying a lot of pain, taking all sort of efforts to make everything happen in the best possible manner. So all of you deserve a lot of big round of applause for yourself, I guess, right? Thank you very much for inviting me here. I'm with all of you. I wish, and I really, I'm dying to see that you all are going to bring more and more global experts to interact with all of you, and they are going to put an impact upon our students, and our students are going to be motivated in the right manner, and they are dedicating their full time, 24/7, in something new, which is going to make their life a different life than others. Thank you very much.
Thank you very much, sir. We are deeply grateful for your invaluable and visionary address. Your presence and encouragement inspire us to strive for greater insights. It is now the moment to make this inaugural session truly memorable for our esteemed dignitaries. For this, I would like to request Honorable Vice Chancellor Professor Alok Kumar Chakrawal, sir, to kindly felicitate Professor Ming Fong Lin by presenting him with a Shrifal and Angvastra as a token of our respect and gratitude. Thank you so much, sir. Now, I would like to request convener of the course, Professor, I would like to request Dean Professor Rajendra Mehta, sir, to kindly felicitate Honorable Vice Chancellor, sir, by presenting him with a Shrifal and Angavastra as a token of our respect and gratitude. Now I request Professor Chandrasekhar Gehan, sir, to kindly felicitate our esteemed registrar, Professor A. Sandeve, sir, with her Shrifal and Angavastra as a mark of our reverence. I now request Professor Renu Bhatt ma'am to kindly felicitate our esteemed Dean, Professor Rajendra Mehta, sir, with a Shrifal and Angavastra as a token of our respect and gratitude. Next, I would like to request Dr. Alka Ekka ma'am to kindly honor Mrs. Fenth and Lane ma'am with Angavastram and Shrifal as a token of gratitude. आप सभी का एक बार पुनः धन्यवाद. In the orchestra of life, phosphorylation strikes the rhythm, redox sets the tone, and endocrinology signs the melody. Together creating the symphony of therapeutics. Carrying this thought forward, let us remember that every successful program is the outcome of collective effort, and it is both graceful and essential to acknowledge those who make it possible. In this spirit, I now have the honor of inviting Professor Harija sir to kindly deliver the vote of thanks.
Thank you, Vikas. A very good morning to all of you. Honorable Vice Chancellor, sir, distinguished guests, dignitaries on the dais, most eminent speakers, sir, my fellow colleagues, and all the participants. It is my honor and privilege to propose a vote of thanks for this remarkable 5-day GIAN program. First and foremost, I extend my deepest gratitude to our visionary leader, the Honorable Vice Chancellor, Professor Alok Kumar Chakraval, sir. Your vision, guidance, and unwavering support are instrumental to the success of not only this GIAN program, but also for our institution academic success. We are sincerely thankful to the Registrar, Professor Abhay Randive, sir, for your administrative support and contribution to the smooth functioning of this event. We are profoundly grateful to our esteemed speaker, Professor Lin, from University of Nebraska Medical Center, Omaha, USA, whose insightful presentation and engaging has enriched our knowledge and enlightened our mind and sparked new ideas and discussions. We are thankful for your presence, sir. A special word of thanks goes to Professor Rajan Mehta, Dean of the school and head of the department, all the eminent faculties of the Department of Biotechnology and School of Interdisciplinary Education and Research for their gracious presence and encouragement and making this event successful. My heartfelt appreciation goes to local GIAN coordinator, Professor M.C. Rao, coordinator of this program, Dr. Santosh Kumar, convener, Dr. Ashish Kumar, co-coordinator, Dr. D.K. Pariha, and all the organizing team, research scholars and students whose meticulous planning and tireless efforts have made this course possible. On behalf of our university, I would like to extend our deepest gratitude to IIT Hyderabad and the Ministry of Higher Education, Government of India, for providing financial assistance to organize this 5-day short-term GIAN course. We are especially thankful to the anchor of this program, Dr. Vikas Chandra, Assistant Professor, Department of Biotechnology, for handling this event. Finally, last but not least, I would like to express my gratitude to all the participants for their enthusiastic presence and active participation in making this program successful. We look forward to your continued support and participation. Once again, I would like to thank everyone for being part of this inaugural event of 5-day GIAN program on multiomics. Thank you, one and all.
Thank you very much, sir, for your gracious vote of thanks and for beautifully acknowledging all who contributed to this GIAN course. Before we conclude, let us all stand for the national anthem and pay homage to the pride and glory of our nation.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल गंगा विंध्य हिमाचल यमुना उच्छल जलधि तरंगा तब शुभ नामे जागे तब शुभ आशीष मांगे गावे तब जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे।
this, we conclude the inaugural session of the GIAN workshop on multiomics, crosstalk of phosphorylation, redox, and endocrinology for therapeutics. May I now invite all to join us for high tea, after which the technical session of the workshop will commence. Thank you so much. हेलो, पीछे वाले स्पीकर से आवाज आ रही है? हेलो, हेलो, आ रही है?
There are two major benign prostate hypertrophy (BPH). So you can see when your grandfather, you wake up in the midnight, often usually caused by the benign tumor we call the BPH, benign prostate hypertrophy. So this the male has the major problem. So because of my research in prostate cancer for the last 40 years, I have a joint appointment we call the courtesy appointment in the Department of Surgery, Urology Division. And because also my research, I have a joint appointment in our Cancer Institute. So you have any question, any issues you'd like to, don't feel hesitate, send me an email. So that's what I put it down, my email is here. So as Vice Chancellor mentioned this morning, very important is your own thinking, not the technique.
Excuse me, sir.
Okay. Microphone. Okay. Working. Hello? Hello? Okay, can you hear me now? I don't know whether the microphone is working or not. Oh, YouTube. Okay, so I still need to wait here. Okay. So this is my email address here. Any question, please send me an email. In general speaking, I do reply to email very well. Okay, we are sometimes become the email addiction. We check the email whenever we are possible. Yeah, so that is what briefly my background. Whether I can pull out. Okay. Yeah, so that is what briefly my accomplishment in my 40 years of research. As this morning, the brief introduction, I have over 120 scientific publications, plus 9 book chapters so far. I have 3 US patents, plus a lot of submissions, but one I did not go through, did not finalize because the money issue. Now our UNMC changed the policy. When you apply for a patent, you need to find a company willing to support it before you can submit. The university will not like to pay for it now. So anyway, it's really, but anyway, try to say when you have a patent application, you can tell yourself, you do have some little bit of different idea from others and a little bit of innovation, a little bit of crazy idea, I will put it this way. So therefore you can get your patent. Okay. I have been invited to over 120 presentation, either nationally, internationally. I have been reviewer for 130 review panel, including in the US, we call the NIH. Oh, and it's NIH, not NH. NIH is typo. It's a major funding resource, National Institute of Health, and the Department of Defense or the VA, Veteran Administration, or the CDC, Center for the Disease Control. Those are the US major federal agency. I have also reviewed one proposal for Canada, okay, or the Belgium, Dutch, France, Spain, Taiwan, because I came from Taiwan, so I review a lot for them. You can see I also review for Italy, Spain, so and so. So I remember one of your faculty member was in Italy. So actually I did review one proposal for Italy, for the country. So the key I try to say is that as Vice Chancellor this morning mentioned, I do have some experience internationally. Although I don't know every other country very well, but at least I know what their function. I can provide you some, if you have a question, I can try my best. And if I cannot, I can try to find the answer or find my colleague to provide the answer for you, okay? So don't feel hesitate when you have questions. As you are a student, it's a time to ask. All the science, I can tell you the truth, all the science come out from your mouth. You don't know it, you ask, you learn it. You don't know it, you don't ask, you never learn it. And you can say, yeah, I can Google it, I can try to find the answer from website. Yes, but you got to find a reliable source of the website. Not all the websites provide you the correct answer. Okay, so we need to have a judgment call. We need to understand which information source is reliable. Okay, because we know there are a lot of fake news in the internet, a lot of fake information. So we got to learn all the single step. So I have also been served reviewer or evaluate for the faculty promotion. I think I don't even remember how many now, or even review the PhD dissertation. Yeah, so all the possibility, all the necessary, I would do the best to provide. It's one kind of our service as a faculty member. Service is one of our requirement, and as not only a requirement, it's also as our nature. We need to provide possible service, not only scientifically or even socially. I even go to presentation, talk with community, how to do the cancer prevention, prostate cancer especially. Yeah, because we need to educate the community people. We are not only educate the scientists, the students, we also need to educate, teach them how to take care of themselves. Okay, so I did go to talk with community people. So that's what actually some of the graduate students in my department said, Dr. Lin, are you going to change your job from professor to a PR person, public relationship person? I say, yeah, maybe I can have my part-time job. Okay, so you got to learn when you talk with the community people, they are different because they don't have so much scientific background as you are or as we are. Then you need to use their own language to talk to them. It's one kind of training too, one kind of idea. Yeah, regarding my publication impact, I found out actually only this is a Scholar GPS can identify where I am. So they have actually given me the 2 ID indeed, because I was in USC, University of Southern California, for 7 years, then transferred to Nebraska. So actually, sometimes the Google search, they're confused. But this is one come out, give me the best. So yeah. So luckily, I have an H index. That means what people cite our publication. So the impact H index. Usually they consider over 40 is a decent score, and we are actually have a 56. Not bad, not bad. I can proud myself. Okay, so far myself, that's my self introduction. I try to tell you what is my background, my qualification. So I think I'm qualified to talk this topic, very broad topic, say the multiomic. Okay, multiomic. So why the multiomic? Yeah, we will going to focus on the protein phosphorylation, redox, and endocrinology. The goal is for therapy. The goal is for therapy. All what we are learning, the basic science, I can tell you is how we can learn the basic mechanism, then apply them to improve to our health. Okay, to improve the quality of our health. Everything what we are doing, all the bioscience, life science research, all goes to this way. How to improve our life quality. So as I mentioned earlier, we're talking about the protein phosphorylation because the protein does not function as a neck protein. It got to be modified. So they can be modified by phosphorylation or glycosylation. By glycosylation, it's other pathway, be honest with you, it's a little bit of not so well established as the phosphorylation. We know the phosphorylation better. We know where they are going to put the phosphate on. So it will be a well-established systematic system we can study it. Then the redox, as I mentioned earlier too, when I was a student, I think the radical, free radical, maybe just a gimmick. I even did not believe free radical. But it's become a major regulation, and even our lab heavily involved. Okay, so free radical become a very important one. So the endocrinology is some sort of more classical term. We don't mention about that too much now, but it's actually involved. We can involve in metabolic. We call the metabolic analysis, metabolic. Right? Why a male is a male? Why a female is a female, for example? It's regulated by the steroid hormone. Yeah, it's endocrinology, steroid hormone. Male regulated by androgen, female regulated by estrogen. But does female contain androgen? Yes, no? Yes. Yeah. So estrogen actually is product from androgen, right? Yes. Just after remove one methyl group, then androgen testosterone become estrogen. Okay, so all this biological linkage, how can we say they are the independent pathway, 1, 2, or 3? It's actually interact. They're crosstalk. So that's what I mentioned, crosstalk. So the endocrinology can regulate phosphorylation, or vice versa. Phosphorylation can regulate endocrinology. So the steroid hormone biosynthesis actually regulated by protein kinase A, cyclic AMP dependent protein kinase, we call pKa. So it's vice versa, they interact, crosstalk. So the redox, similarly. Okay, so to the end, I will give example. But in the class, so this is 1 week. So today I'm going to focus on the basic, some sort for students, they don't have the background in biochem signal transduction, then I some sort of give you the background. For those who have background already, we just try to refresh your memory, refresh your mind in more detail. So then tomorrow we are going to talk about the redox, free radical. Then Wednesday we discuss about the endocrinology, the steroid hormone signal pathway. So for the steroid hormone signal pathway, we're talking about the androgen, estrogen, the progesterone, the glucocorticoid hormone. We all think about gene regulation. But is that really the case? All the steroid hormones through gene regulation, turn on the gene, shut the gene? No. Actually, now we know non-genomic action plays an equal role as to the gene regulation. So steroid hormone does not need to regulate the gene expression. They can through, for example, the calcium. They can regulate the calcium for signal transduction pathway. So that's what we are going to discuss on Wednesday. Then the Thursday is some sort of a summary. I'm going to summarize. We're talking about the targeting therapy. As I mentioned, the study eventually we are going to do is to improve the quality of our life. So if we targeting one disease, we understand how they crosstalk. We call the mechanism, pathogenesis, or the pathogenesis. Then we can target to those key molecules and design the drug for therapy. Okay, so for example, one of the growth factor receptor we call the HER2, EGLR2, HER2 new, right? So in breast cancer, in ovarian cancer, about 10% of patients has amplified, gene amplification of HER2 new, about 10%. So when we identify a patient has HER2 new, HER2 amplification, then we could quickly use the antibody we call Herceptin for targeting therapy. So it's very powerful for breast cancer. Very, very powerful treatment for breast cancer. But interestingly, in ovarian cancer, they can have also HER2 amplification, duplication. But the Herceptin treatment efficacy seems not so great as in breast cancer. Why? Why? Okay, so all those questions relate to the molecular mechanism, signal transduction pathway. We can really get into it to understand it. So, but for the sake of the time, we are going to, sometimes will overview what's happening and give you some example for the targeting therapy. And I will tell you to find out in the 4C, for example, I will give you an example that what we are studying. A protein we call the P66 protein. It's a protein without a function, but turn out it's going to mediate the cell hormone, the free radical, the growth, the tyrosine phosphorylation, and not only involved in the carcinogenesis, actually also involved in aging. How long you are alive? The aging. Okay, so that is a longevity gene. Shorten or prolong our life, I cannot tell you either. Because depending on your environment condition in the mouse study, the knockout mice in the cage, the lifespan is prolonged 30%. It's very significant. So that means you can live, you can almost average 70 years, but you don't have the P66 gene, you may be prolonged for 30%. It's really a long time, 30%. But that is for the mouse in the cage. But you then put the mouse, the mouse, in the natural environment, then the lifespan actually shortened. So it's also being called as a selected gene for the life, because this protein not only involved in the free radical, but also involved in the fat metabolism. So in the winter time, we require fat. So then, if the mouse, the gene being knocked out, in the winter, they cannot survive well. Okay, so a lot of factors. We're talking about this gene doing this function. We talk about that gene doing that function. We need to be careful under what condition. Under what condition. So I'm going to tell you a lot of basic knowledge, a lot of basic science, but I'm not going to tell you what to do. As what the Vice Chancellor mentioned earlier this morning, is how those Asian scientists, they can do the science, make so much discovery when there was no tool, no instrument. How they make the discovery? It's in here. Your brain. Okay? We need to be creative, be challenged. So some students answer to my question, say, oh, we just put into the machine, we just have a machine analyze that, analyze this. But when you don't have a machine, then how are you going to do? And the result come out from machine, for example, the MMR, or you have a lot of data, right? Those kind of, so how are you going to decide which one data is the one you should go for it? There are a lot of pathways. Proteomics, you come out a lot of peptide, a lot of protein. You can link a lot of pathway. But which pathway is really the critical one? How are you going to decide? The machine won't decide for you. It's yourself. Okay, so very important, understand the basic, those kind of concepts. Then you make your judgment call. So for example, when we studied the P66 shift, actually, Dr. Santosh Kumar was involved when he was in our lab. So this protein initially was discovered as involved in apoptotic pathway. Oh, by the way, I really don't know what your background. So how many students have biochemistry background? How many? Most of you? So biotech. So biochemistry major. Okay. So P66 shift protein, for example, was initially identified as an apoptotic protein. But in our lab, actually, one of my a former associate, a very smart guy, he came to see me. Initially, he was involved. He said, Dr. Lin, can we study this protein into cell growth? So we know cell growth, apoptotic, are the two different apoptotic pathway. He said, yeah, all the people study apoptosis. Why won't we study cell growth regulation? I said, are you crazy? He said, no. He said, okay. I said, do it. That's it. Okay, so you got to think something different. Then you got a new project. And the project we actually study continue going on even now. We found out this protein is more than what I thought at the beginning. Yes, it's involved in the regulation a lot more than what we know. It's because it interacts with the mitochondria. Cytochrome C can have RS production and it can do something else, involved in the lipid metabolism. So this protein turned out more complicated than what I thought. And I can tell you our newest data, this one even involved in the carcinogenesis. Yeah, involved in the carcinogenesis. Not just we can elevate its expression, the immortalized cell become transformation. So you understand the immortality, normal cell, I would say normal cell become transformed. Okay, so that means the cell become transformed, become malignant cell. It can grow continuously, can grow in the soft agar. Okay, so become more than what I understand. I can tell you that. So Friday lecture will be by two others faculty members. So they are going to introduce their own. Okay. So far, any question? So this is one I'll try to tell you why. Because, for example, this is very simple talking about neuron transmitter. So the easiest from hypothalamus, we can have a factor release. This is endocrinology. Okay, the factor release circulation, right? Then they will through all the different pathways then to regulate our physiological function. This is in the normal human, normal animal, or we can name it. So this involved in the endocrinology, involved in the signal transduction pathway here. So that's what we call, this is the first messenger, endocrinology. It's just circulating in the body. So when they hit to their receptor, okay, specific receptor, then they can target and go inside the cell. Then inside the cell, we call the second messenger. So all the body can be regulated. So complicated, so complicated. So there got to be a way how we can have the judgment, how the cell can make a judgment call, go to this direction or go to that direction or with the balance, or why a male will synthesize the testosterone, androgen, but the female will go for next step to estrogen. Because they're all from the same precursor. What is the precursor for steroid hormone? Cholesterol. The only source of steroid hormone is cholesterol. So why the cholesterol can be processed through which step they will start? Okay, they will start to get, say the first step will be progesterone. Then we go down to the progesterone and the glucagon hormone will be the same thing. And then they will go down to the androgen, male hormone. Then they will go down to female hormone. So sometimes when I give my lecture, I say, you know, I am very impressed by those philosophy people. They would propose in, say, in Christian, okay, in Bible. They're talking about, say, female is come out from a rib, one rib or a male. But estrogen indeed a product from androgen, testosterone, male. Yeah, they have no science. As we know now, at the time they did not have science. But how come they do have that description? No, we are not talking about the religion. We are talking about the science. So that's what the Vice Chancellor mentioned today is very important. There was no instrument It's purely depend on our brain, our thinking. How can they have so creative? Okay. Okay. So the multiomic is actually linked the gene, the protein, and the metabolite, all the regulation together. So we now have what people call the genomic, epigenomic, or we call the proteomic, or metabolomic, or transcriptomics, or metabiomes. So, for example, the transcriptome we used to call functional genomic. So when the beginning try to study how the gene expression, we actually have the membrane, we spotted, we identify the mRNA. Okay, so we call the functional genomic is the mRNA. But now they expanded to transcriptome. And then metabolomic is just metabolite, but essentially regulated by the endocrinology. Then the proteomic, we can actually expand to have phosphoproteomic. Okay, it's protein phosphorylation. Or it could be glycomic, protein glycosylation. Or we can call glycomic, it's a redox, then etc. You name it. Whenever you can create one, you can name it. Nobody is going to say no. Okay, so all this depends. So it's essentially a combination. But as I say, we are going to focus really on the proteomic, how the phosphoprotein, then the redox, then endocrinology, how they interact in this pathway. Then we actually did involve in the metabolome analysis. Actually, the data can come support redox. We did have the data from metabolome to support the redoxomic, okay. And we have the data to show we call the functional genomic compared with currently. So metabiome, microbiome. What is microbiome? The microbiology, right? So in our body, in our colon, we do have microbiology there. Yeah, those microbacteria, everything over there. Are they important? Or we think those are not important? Initially, we don't think those are important. But actually, they are very important. It's regulate the microbiome in the body, actually regulate our immune system. Okay, not only regulate our immune system, actually regulate patient drug resistance. They find actually some patient resistant to certain chemical reagent, right? So what they do, and some are more sensitive. So what they do is they transfer the frequency from those sensitive patients to the resistant patient. Then turn out those chemo-resistant patients become chemo-sensitive. So the microbiome actually play an important role in the body to regulate our drug resistance. So the immunology, now we know the drug resistance. So that's what one of the proposals is, can we treat the cancer patient, those resistant one, with a frequency from a sensitive patient. So we just transfer a little bit of frequency from those patients because they contain different microbiome. So then the patient will become sensitive. We never thought of that, and we did not believe it. But now those are the case. Okay, those really the case now going on. So it's really, so that's what I mean is so much going on. So we try to focus mainly on the protein level, okay? So this is what we are talking, the first one section. So we're talking about the hormone, the signal pathway. So this is a very simple, just a basic how the molecular biology, the signal transduction pathway, how is a hormone bind to the receptor can transduce the signal to so many pathways to regulate the ion transporter, the metabolism, the gene expression, the same movement. So how we can regulate? So how is it's actually essentially can be the same receptor doing the different pathway. How can we do it? Okay, so that's what we need to understand, and then we understand the hormone can be as a peptide. Okay, so can be as a steroid, can be as amino acid derivatives. So those are the very basic biochemistry. As I said, for you have no background, we try to give you some idea. For you have background, we try to refresh your memory, all this regulation. So again, then how the cell-cell communication? So for example, there can be a direct contact, cell-cell contact communication, or could be a paracrine communication. One cell secreted the ligand to interact with the target cell. So this is a paracrine system, and this is what we also call the ghasidas system. Or it could be the autocrine system. Actually, the cell secreted the ligand and react to their own receptor. So most of the case, this is in the cancer. Okay, those are in the cancer. So the autocrine system also considered could be possible intracrine, could be the same this cell and then another cell adjacent to it. They can be crosstalk. So essentially, this is the cancer cell mainly. The normal cell injury that would not happen. Then they do have the endocrinology. Then this, as I mentioned, this is a classical term. Now we mention to the metabolism, continue to be the metabolism. The SNAP interaction, okay, the neuron junction. So this would belong to the neurobiology system. We are not going, in the signal transduction, we don't talk about this too much because this is a focus by the neural people. They are very fast, very rapid. But do they involve in the signal transduction? Yes, they do. Because the junction can also have protein kinase, they can have the protein phosphatase. So also regulated by the phosphorylation. But their regulation pathway, the speed is much faster. So we're talking about, say, for example, cyclic AMP, cyclic GMP. They are the same, it's through the protein kinase, right? But the mode of the regulation is slightly different, and then the speed very dramatically different. Because, for example, cyclic GMP, PKG could regulate our heart attack. But the cyclic AMP, in general speaking, regulates metabolism. So the heart attack we know is a very rapid process. It counts by minutes, seconds. But the cyclic AMP, it could be by days, hours, very slow. Okay, so yes, there are signal transduction. The junction also regulated by protein phosphorylation, the kinase and the phosphatase. Yeah. So this again, I describe where the receptor, where is the ligand. So for example, the receptor, they are on the membrane, or the receptor in the cytosol, or the receptor in the nucleus. So they can have different localization. So the second message, in general speaking, we focus on the receptor on the plasma membrane. Okay. So the steroid hormone allowed, actually the receptor could be initially at the cytosol, then translocated into the nucleus. So those are the ligand, lipid-soluble ligand. So they are the way. Then the steroid hormone receptor, for example, it just localized in the nucleus directly. But the androgen receptor, estrogen receptor, for example, they can be in the cytosol and then translocated. Or some of them may be already in the nucleus already. Okay. But then we know actually now the steroid hormone receptor could be also on the membrane. Can be also on the membrane. Yeah. Okay, so this is another example, another slide to show you what the intracellular receptor and what is the cell surface receptor. So intracellular receptor usually is a hydrophobic hormone. Then for the cell surface is a hydrophilic hormone. So the hydrophilic hormone cannot be delivered through the plasma membrane. So the receptor is on the surface. But for the hydrophobic hormone, they can be translocated inside to the nucleus, inside to the cell. So the receptor is intracellular. Okay, so these are the basic difference. So this is, I prepared the detailed slide. You can go through it when you have, if you have any question. I understand there is a little bit of language barrier between me and you guys. Okay, actually, to some people, maybe very substantial. But we got to understand now all the science is global science. Yeah, it's global science. So we got to, so I try to understand your English, you try to understand my English. So in a way, I prepared the detailed slide. You can, if you cannot follow my language, then you can follow the slide to go through it. Okay, I think they will make it available to you guys, right? Yeah, because they do have the video. I don't know. Okay. So this is talking about the hormone binding. What is the cellular response versus the fraction, the receptor binding. But then I try to tell you is they actually, even a textbook can have a typos. Okay, don't believe all the textbook, they have a mistake. They are correct. They do have a typo. So this is what I try to mention why I still use, I know the figure is not correct, but why I still adapt it, I save it here to present. I try to explain to you, yes, this is what I mentioned at the beginning. You need to find reliable source. The source, even the textbook, it still could be a typo. It still could be a typo. Okay? So again, this is talking about the membrane receptor. So there are two different kind of ligand. Got it? Receptor or the enzyme link, or then the cytokine receptor, or then the G protein couple receptor. So they do have different, the receptor could be functional, could have their own enzyme activity, or the receptor may not have an enzyme function, but it depends on the adaptive protein to do the function. Okay, so these are the different kind. We will focus a lot on the GPCR, G protein couple receptor. Yeah, G protein couple receptor. This is one of the major ones. And also the cytokine receptor is also very important. Essentially, they are important, but the GPCR is a lot, G protein couple receptor. For example, we can say about 30% of currently drug on market, they actually targeting to GPCR. And this pathway, okay, it has attracted so much attention. The Nobel has met so many people by studying the GPCR signal pathway and then receive the Nobel Prize. That becomes the Nobel laureate. So many of them. Yeah. So then that is the next one is talking about how when the receptor on the membrane, when the ligand bind it, what will happen? They can, like a transferrin receptor, they can recycle. The EGL receptor, they can recycle, or they can go to digestion. Or the alpha-2 microglobin, they can be just go to the lysosome and go to degrade it. So all depend on different kind of ligand, the receptor, they can have different regulation. So these are the three major pathways. Then we are talking about, we have all those classical regulation, endocrinology, but now they come out with estrogen. Yeah, estrogen. So that is a cell come out with a small microvehicle. Okay, so this is a microvehicle coming out from the cell. Then they can contain a lot of materials. They can have protein, they can have mRNA, they can even have DNA. So in classical, in our research, we thought the estrogen is maybe a contamination. Cell contamination, we did not think, we did not believe those estrogen has any function. Until the 1983, yeah, different labs, they come out and say, yeah, it's not the case in the red blood cell. actually the exosome is functioning. So the study going on, but even now, still a lot of confusion. But the bottom line is the exosome actually is the same user receptor. Okay, use a receptor to do their function. We will discuss it to the end of the today. Okay, so this is briefly what we're talking about, the basic biochemistry, how the ligand, the circulation, how they react with the receptor to the function. Okay, so any question? Any comments? Okay, so this will be the second messenger. What time is done by today? By this hour. I'm a little bit confused with the schedule. Do anyone have the schedule? Oh, I do have. Oh, no, not this one. Yeah, I think I have one. They give me one copy. I got it too. Thank you. Okay, so we need to finish today by 1 o'clock, right? Okay. No, by 1 o'clock, then you have a lunch, then we come back until at 2. Okay. So we just try to go through a little bit about the second messenger. Okay, so we're talking about the second messenger. So the first messenger is outside endocrinology. The second messenger is one after the membrane, after the receptor. Okay, so they generate the messenger. So now classical we have the cyclic AMP. Then subsequently we have cyclic GMP. Those two are the base study. Now we know they have a cyclic CMP, or even they have cyclic UMP. So essentially all the nucleotides they can serve as a messenger, second messenger. So the cyclic AMP was through the PKA pathway. Then the cyclic GMP actually have their own cyclic GMP dependent protein kinase called the PKG. So this is a PKA, this is a PKG. But the cyclic GMP can also interact with PKA. Okay, can also interact with the PKA. Then the cyclic CMP, they usually interact with the cyclic GMP kinase 1 and also protein kinase A. So it's some sort of interact, not very exclusively. So this could be the biological function. This is the summary slide talking about the major three. Then the cyclic UMP, that's what we did not mention. Is we don't know much. It's mainly in prokaryote. That's what we understand, but we don't know too much. So as I mentioned here, the role is much less known. Okay, we know better cyclic AMP, cyclic GMP, and the cyclic CMP can also function involved in the eukaryote cell, but a lot also in the prokaryote cell. Yeah, so it all depends on this research. The CMP or the UMP will depend on your discovery. To me, I don't think I can see that discovery, but it's your time. You are interested, you should try to figure it out. Okay? So then we're talking about, those are the, so we can see the protein phosphorylation, mainly as a serine, threonine, and tyrosine. So classically, we always think phosphorylserine is a major one, and the phosphotyrosine is only, say, 2%. But remember one thing, this is depend on the acid hydrolysis. How we determine amino acid composition, how we determine phosphorylation, all depend on acid hydrolysis. 6 normal hydrochloric acid hydrolyzed. And turn out phosphotyrosine is very, very unstable. Okay, I can tell you even phosphotyrosine amino acid water solution, you keep them in the refrigerator, it auto hydrolyze. It auto release hydrolyze. So when we perform the experiment, we always need to have phosphotyrosine as a standard marker. Then unfortunately, because there are thermodynamic entropy is so low. Okay, so the entropy is a kcal is about -7 to -9 kcal. But phosphoserine, phosphoserolin is about -1, -2 kcal per mole. So this is much unstable. So every time when we use a standard solution, we need to add this powder, fresh powder in to prepare the fresh one. Otherwise, you may not see the signal. Okay. So we need to understand for the biochem, sometimes people say, why it's so complicated? Is we need to understand a lot the basic chemistry, not only what they are doing, we need to understand their basic chemistry. So when did they discover? You could not believe in the 1980s, we know the protein has phosphate group already. Then in the 1950s, we discover, we means scientists discover kinase. Then the kinase, when they find out first, actually, histidine phosphorylation was discovered in 1962. So you never heard about histidine phosphorylation, but it was discovered in 1962. So the reason is we don't study it much is even the phosphohistidine is even much unstable than phosphotyrosine. We say phosphotyrosine is -7 to -9 kcal per mole. Then this is even much, phosphohistidine much unstable. Okay, so acid hydrolysis is essentially impossible. So the study is so different. We have more study on tyrosine phosphorylation. This was discovered until 1980s by Dr. Tony Hunter at Salk Institute in California. But be honest with you, tyrosine phosphorylation was discovered way back in World War II by German scientists. German scientists, they studied protein tyrosine phosphorylation during the World War II wartime. So the question always in our minds, why? Why they were so interested during the World War II? The resources is so scarce. Why those scientists had nothing better to do, study protein tyrosine phosphorylation? So this actually, Dr. Hunter, Tony Hunter, dig into the library and then find out in the World War II, German scientists have been studying the tyrosine phosphorylation. And in the real world, we actually get into it until 1980, until we find out oncogene protein, growth factor receptor, they are the tyrosine kinases. Then the study means some sort of bromine. Okay. But why German scientists, they had the sense, had the taste in the World War II time zone, studied protein tyrosine phosphorylation? I don't know either. My understanding started from 1980s, but those are the history we take back the time. So now we know actually there are 9 different phosphor residue. Okay, you can see how many. It's not only serine, serolin anymore. There's 9 already, and the 4 types of the phosphoprotein linkage. Yeah, 4 types. So it's more complicated than what we understand now. But of course, we cannot study everything, so we can pick and choose whatever you are interested in, and we consider more important one. So this is another one, tell you what the difference. So in the 1980s, as I mentioned in the first previous slide, in 80s, we know protein has phosphate already. So that's due to the phosphorylase. Okay, so phosphorylase. And now we study the kinase. So what's the difference between a phosphorylase and a kinase? What's the big difference? No, kinase use energy, usually ATP. Phosphorylase, no, they just use phosphate. But the same, those 2 enzymes can put the phosphate group onto a protein. Okay, so the main thing different I circle here. Yeah. The kinase require ATP in the energy source. But the phosphorylates, they don't. They don't need the energy source. They are the major difference between the two. So this is one is some sort of modified. Yeah, if we use acid hydrolysis, we can see most of them are phosphoserine, and then phosphoserine, phosphotyrosine a little bit. But use alkaline hydrolysis in opposite, actually the phosphotyrosine will increase depending on the protein, came from 1 to 2% go to up to 10%. Okay, 10%. It's more stable in the alkaline solution. But phosphoserine, they actually not so stable, so the percentage going down. Yeah, percentage going down. So that's what I put down the note in the red color is phosphotyrosine more stable in alkaline solution than in acidic condition. Okay. So that's what I try to emphasize. That a lot of students, a lot of people think phosphoserine, phosphotyrosine, the phosphotyrosine is so tiny amount. It's just the detection technology. So why yes, instrument is good. We need instrument to advance our research, but we also don't try to be misled by the instrument. Because instrument is limited, has a limitation. Okay, instrument has a limitation. Yeah. So this is the GPCR, G protein coupled receptor. It's required the GTP binding, but not the ATP. It requires the GTP binding. So that's what we call the GPCR, G protein coupled receptor. They can have through the GTP bind to the alpha subunit, and they can have different diverse signal pathway. Yeah, so the main thing we study here is called the so the PKA pathway or the PKC pathway. What is the PKC pathway? It's dependent on the calcium, because the calcium, so we call PKC. Okay, the calcium. So the PK is a cyclic AMP dependent protein kinase. We call it the PKA. Yeah. Okay, so they do have different kind of alpha subunit. And not only that, they do have different beta and gamma subunit. So that caused the GPCR study, it becomes so complicated, so confusing, and actually involved in the level regulation. So I just mentioned, say, over 30% of currently prescription drug against the GPCR, G protein coupled receptor, because they do have alpha, beta, and gamma subunit. So in 1994, because of study of this pathway, Dr. Gilman and Robert, they received the Nobel Prize for the signal transduction of GPCR, cyclic AMP GPCR. Yeah. Then this is 1994. Then 2012, another Nobel Prize. So you can understand how important the pathway. And not only that, we have other pathway. We have another award coming. Okay. So then this is GPCR, G protein coupled receptor. We call it the 7-span protein, because 1, 2, 3, 4, 5, 6, 7. 7 spans through the proximal bandwidth. So we call the 7-span receptor. So they require the GTP. The GTP binding is activation. GDP binding is inactivation. Okay. So the loop here interact with the G protein. Then here is the serine phosphorylation. They were regulated by serine phosphorylation. So then the GPCR also involved in our smell. Yeah. The odorant or the small molecule through here or the protein TSH, all this is again GPCR pathway. So this is another Nobel Award in 2004. They discover how the smell, how the regulation of how we smell is a G protein coupled receptor. Then in 2021, yeah, the temperature, our touch, is also the same through the G protein coupled receptor. So now you can understand how complicate the system for the G protein couple receptor. Okay. So my student used to tell me, say, yeah, if I can identify a novel pathway, I can publish a paper in a high impact journal. I say yes. If you can identify a novel pathway and all the regulation, novel pathway, novel regulation, you can publish it in a high impact journal because it's so important in the signal pathway. So this again is a refresh. We sometimes try to repeat about it. Okay, so this is a GTP bound active form, GTP bound in active form. Okay, reiterate. Then what is alpha subunit, beta, and the gamma subunit? Then this tell you again how the cycling, how the recycling, when the GTP bound, then the GTP bound alpha subunit will be separated. Then we go to the activate the adenyl cyclase, then have cyclic AMP. So we're talking about the chlorotoxin. Okay, in the tropical area, the chlorotoxin is one of very important one, a major etiology for the diarrhea, especially for kids, because the chlorotoxin will modify the alpha subunit. So the alpha subunit will be activated, will be remain active constitutively. Then we got the dehydration. We got diarrhea, we got dehydration. Especially for newborn baby, if we are not careful, there's a life-threatening issue, the chlorotoxin. Okay, so it's happening a lot in the tropical area. So the GTP bonds then will become inactive. So this is a recycling pathway. So this one gives you much clear how the chlorotoxin and also pertussis toxin on the diarrhea issue. So I just mentioned the chlorotoxin actually activates the enzyme, keep the enzyme continuous activated. So then we got the problem on the enzyme will be on constantly. But on the other pathway, the pertussis toxin actually is inhibit the G protein. So have the similar result is the protein become active. Okay, the protein, yeah. So that is the same problem, the same result. We have the cyclic AMP problem. Okay, so the toxin, why we, as I say, we study the biochem pathway, we can understand some of the mechanism of the disease, the pathogenesis, then we can develop the drug to inhibit the enzyme to cure the disease. Okay. So this is a cyclic AMP pathway. So methylxanthine actually can upregulate cyclic AMP. So what is methylxanthine? Where they are? We drink the coffee, we drink the tea, they have methylxanthine. Okay, compound. So they can activate the protein kinase because they inhibit the protein diesterase, cyclic AMP phosphodiesterase. Okay, they inhibit it. So this is here, cyclic AMP phosphodiesterase. So suppose this enzyme will deactivate cyclic AMP, so then the signal can go down. But because this compound, the caffeine or thrombin, can inhibit the diesterase, so the cyclic AMP remain. Then the enzyme is constant, so that we can wake up. That by caffeine, one of the mechanisms. We wake up by drinking the caffeine. We're not going to sleep. So because the caffeine, all this actually inhibit. So when you drink too much coffee, so what is the side effect? Except you might keep yourself wake up all the time, but what else? When you drink enough coffee, when you drink, say, 1 pot, 10 cups of the coffee every day, okay, for a month, your hands start to shake. Try to shake. You cannot control it. So when I was a postdoc, I need to study, so I need to experiment so hard. So I drink the coffee continuously, 1 pot per day by myself. Then my fingers start to shake it. I cannot type. I cannot do experiment. So I was shocked. I said, what's happening to me? I said, oh, could be the caffeine, because I remember this mechanism. I said, oh, could be the caffeine. Then I quit drink coffee. Bingo. So sometimes it's good to be a scientist. You understand what could be the potential problem. Yeah, I quit to drink coffee. My hand stopped shaking and immediately. Okay, so that's what. Yeah, so I think Dr. Kuma Santosh came here. The boss is here. And maybe we need to have a break. Oh, it's 1 o'clock. Yeah, so we can stop here. Then you guys can have a break, have lunch, and then we come back. We can continue. It's 1 o'clock. Okay, any question? Quick.
You told us that phosphohysteine are very unfixed. So could we say that they are used in transient activation of proteins?
Which?
So you said that phosphohistidine and phosphotyrosine
Ah,
are
yeah.
unstable.
Yes.
So could we say that they are used in transient activation, short-term activation?
Yeah, they do see it. It just, as I said, phosphotyrosine is unstable. Yeah. Right? It's not, so we can actually detect it with acid hydrolysis. So if we try to see it clear, we use alkaline hydrolysis. But we still can see it. And luckily, the phosphotyrosine antibody is commercially available now. So it's much easier. You can purchase antibody and then it's very specific. They do have antibody to phosphoserine, phosphorylserine. But turns out the phosphoserine, phosphorylserine phosphorylation so much, so abundant. When you run a gel, you see so many bands. You don't know if that's really the case or they are just non-specific. Okay, so we don't understand the case. But phosphotyrosine is now very clear. The antibody is very, very good. Phosphohistidine antibody, I don't see the commercial availability. People did try to produce it, but had a difficult time. So the study phosphohistidine, that's what I mean. It's much slower, much, yeah, that is the reason. You need to have a tool. So we cannot, then what is when the tool become available, especially when phosphotyrosine antibody become available, then the study just boom. But at the beginning, there was no antibody either. So we need to use phosphotyramine for immunization. But turn out phosphotyrosine, we can immunize too. That's people worried, did not think that it could be the case. Yeah. Okay, so that is how we can improve it. Then, as Vice Chancellor said this morning, the same is we need to think how we can improve it. So initially we thought phosphotyrosine unstable, not good for immunization. So people develop phosphotyramine, so similar structure, then to generate antibody. But then they find out, oh, no, actually phosphotyrosine is good as phosphotyramine. Then they use it. So it's how we are going to move forward. This is a science that, so what we call research. You remember the word research, search, right? We keep rebranding the wheel continuously. When you have better tool, you have better idea. So that's what so many people study cyclic MP, PK pathway. That's what I mentioned. And why the Nobel Prize committee awards so many awarded to the same pathway? But they found out this pathway involved in so many functional regulation. Yeah. Okay, so that is really depend on how in here. And then also you think you need to do it. Okay? Okay, thank you. Yeah. you have interest in how you are going to apply to US the program in the US. Okay, so my suggestion is discuss with your advisor, with your professor, who has experience to US. So, for example, Dr. Kumar here, he has been in US for many years. He knows some of the process, so he could make a suggestion, recommendation, which program you are interested in, and then which university could be good to apply for it. Because every university, or even within the university, each program has different requirement, specific requirement. So, for example, at University of Nebraska Medical Center, we have a unified program. You can apply for it, but within it, they do have a subgroup. So, for example, we have biochem group, we have cell biology program, we have cancer biology program. So, depending on which program you are interested in, and each program might have their specific requirement too. So I cannot tell you exactly what we have, what our requirement. So you need to check into it and see what their requirement. So one thing, as I mentioned, is the most important is your undergraduate score, your GPA. Okay. Say for example, if the GPA 4.0 is a 1 to 4, then you have a 3.9. That's outstanding. That's very good. But you have only 3. Then that is not great. Then another thing is your reference letter, recommendation letter. You got to have very strong recommendation letter. The professor who knows you well can write down very specific, very highly recommendation. Don't just say, oh yeah, you say you took the class and you did something extra, then the professor actually like it, and he knows you very well, then he can very specifically recommend. Put it down, say, yeah, he considers you are an outstanding student, outstanding candidate. Why? Then he can put down very specifically. Then your chance to be accepted, of course, much higher. But only a professor say you are a student in a class of 60, 70, then the reference rate, and then the professor may not know you well, then your letter may not be strong. Okay, you got to let the professor personally know you, then the letter could be more personal and much stronger in that way. Yeah, don't be just very general. Okay, so that will be my suggestion, advice, how to process, how to apply. Then the application now is very slow. Even it's online application, but because the visa issue, everything is so slow, everything is so delayed. So I would highly recommend as soon as possible. If you are preparing for going the next year, you had better to start quickly now. Yeah, usually I think the deadline of application is the end of the year for some university. Maybe can be say until the next January, but for a lot of good university, they might do date by the end of this year. Okay, the end of the year, say December. I remember when I applied to come here to go to USA when I was in Taiwan. I was a student. Then I received my acceptance letter after the new year. So that means they made the decision, everything way before the new year. Then they could mail me the letter, acceptance letter. At the time, I used the mail, regular mail, right after the new year. So that is the case. So it's got to be, yeah, especially for those high competitive universities that you think you're interested to go, then the earlier the better. Okay, have better chance. Yeah. Okay. Any other question before we start? Yeah. Okay. So now we are yeah, so this slide shows how it's a PKA, protein kinase A signal pathway, how it's in the membrane, how the signal come in, the GPCR through the adenyl cyclase. Okay, so the GDP binding, then they have used the ATP to produce the cyclic AMP. Then the cyclic AMP could activate the protein kinase A. So that's what we call the PKA, protein kinase A, or called the cyclic AMP dependent protein kinase. So then that can, yeah, so the binding of the cyclic AMP, then the dissociate of the, they will activate the PKA. The catalytic subunit will separate from regulatory subunit. Then the activate subunit then can go to the nucleus. So usually in here, they can do the protein phosphorylation. They can also go to the nucleus, activate the gene expression. So this is what you can think is the signal pathway, not only in the protein phosphorylation, they can also regulate gene expression. So as I mentioned here, the catalytic subunit, C is the catalytic subunit, can go to the serial signaling or go to the gene regulation. In here, the example is a CREB, CREB, okay, protein. So that's a very important protein regulate in the protein phosphorylation pathway. Yeah, so because the discovery of the cyclic AMP actually was a big story way back in 1971 by Dr. Sutherland. So he discovered the cyclic AMP. Then, be honest with you, this actually moved the whole signal transduction pathway forward. Because at the beginning, we did not understand how the signal is secondary messenger. So because of this discovery, then we move the pathway, and we understand that through the PKA, cyclic AMP dependent protein kinase. But unfortunately, Dr. Sutherland passed away a few years later due to traffic accident. So yeah, some sort of slowed down a little bit because, as you know, he is a pioneer in the field. And when the accident happened to the pioneer, then the rest of the group will slow down their step. Yeah. Then because of this PKA, then we know there's a protein kinase, there's a phosphatase for regulation. So this actually received Dr. Fisher and Dr. Krebs received the Nobel Prize in 1992 on this pathway. We also think kinase, right? We talk about another phosphorylation. One of the reasons, because we know that the patents is much easier to study. As when I studied, actually my interest was studying the protein phosphate. So when I studied the phosphate, Tony Hunter, he is the one discovered tyrosine phosphorylation. He asked me, he said, Rico, why you study phosphate? Why won't you study kinase? I asked him, I said, Tony, why? I know him personally because he is a good friend of mine for staff. So we met and we chat. So I asked him, I said, Tony, what do you think? He said, yeah, when you study kinase, you add ATP and you see the phosphorylation. You know this kinase, right? You have the protein. But then when you study the phosphate, you need to see that the phosphorylation disappears. Then you have a protein. If the phosphorylation won't go, you don't have a protein. So it's the next step. Even it's a very important in integration. Actually, it could be more important. But it's the second step of reaction. Then the study much different. So I say why the sorry. Yeah, so why I think phosphatase could be more important. You know, 2% of brain cell okay, our brain protein, 2%, is a protein phosphatase. 2% of brain protein is a protein phosphatase called carcinoid. So you think 2%, oh, that's a very tiny amount. But you consider how many different proteins and the phosphatase composite of 2%. That is substantial. It's a phenomenon. So in the memory, actually regulate our long-term memory, the phosphatase regulate it. Okay, so it's just a kinase study, much easier. As what Tony Hunter said, you added ATP in, you see phosphorylation, you got a project. But then the phosphatase, you need to wait until it's dephosphorylation, then you got a project. It's much slow. But the importance is equal. Not say kinase more important than phosphatase. Okay, so this is what the yeah. So you can see now again the pathway, but on the PKA pathway, how many Nobel Prize has been awarded? So you can see how important this pathway in regulating our metabolism. So PKA we consider is a regulation of our metabolism. Okay. Then tyrosine phosphorylation is involved in the cell growth regulation. But serine is involved in metabolic regulation. Yeah, we can briefly say that. Because most of the oncogene or most of the tyrosine kinase are either their oncogene protein or the growth factor receptor. Like we know the very clear epidermal growth factor or insulin receptor. Even insulin receptor is a tyrosine kinase. Okay, but then the phosphorylation, serine are involved more in the metabolic regulation. Like a glycogen metabolism is regulated by PKA pathway. Okay, so this very briefly give you the idea. Okay, so this is about the PKA. Then the next one will be the PKC, protein kinase C. So protein kinase C is a regulate, okay, the require the calcium. So that's what we call it the PKC, protein kinase C. It's calcium dependent. But actually, so this we call is the phosphoinositol cascade. It's the activated PKC, serine phosphorylation. So it's required the calcium is very important and the DAG. And some PKC require comodulin. Some PKC does not require. So we can call comodulin dependent PKC or comodulin independent. Okay, so this is a very clear diagram how the G protein coupled receptor can activate the PKC pathway. Yeah, so this what I described in here is PIP2 then go to hydrolyze become the DAG plus PIP3 by the phospholipase C. And they both of them can activate the PKC pathway. Then they do have a GQ subunit. So this is also the phospholipase C. One thing very important is the phospholipase C has alpha, beta, and gamma isozyme. Then only the gamma, PKC gamma, can interact with tyrosine phosphorylation. Okay, PKC gamma. Alpha and beta are serine phosphorylation. But the isoform gamma is through the tyrosine phosphorylation because it contains SH2 domain. So SH2 domain is one of the interacting domain in the tyrosine phosphorylation. Okay, in the tyrosine phosphorylation. Are we okay? Yeah, I know it's so the PK serine phosphorylation a bit different from tyrosine phosphorylation. Tyrosine phosphorylation all depend on protein-protein interaction through the SH2 domain or SH3 domain binding. Yeah, but the serine phosphorylation they could through say the calcium or they might through the DAG or yeah. So it's a different kind of the regulation pathway. So this is the calcium comodulin dependent protein kinase. It's more detailed how involve the chymogloin. It's a protein. So the discovery of chymogloin actually at the time, people thought this could be a totally independent family in the PKC pathway, but turned out it's just a protein can activate a little bit more activity or not. Okay, so quickly we went through the pKa, now the pKc. Now we are talking about the cyclic, those are the cyclic AMP. Cyclic AMP. They can have the pKc, pKa pathway. Now we're talking about the cyclic GMP. So it's similarly, they're regulated by guanidine cyclase, also it's a phosphodiesterase. So the same is they form the correct cyclic GMP, then they could be hydrolyzed here. So like a cyclic AMP, we can hydrolyze this one, cyclic bond. So it can function as a secondary signal. They can have vasodilation, okay? So very important, the cyclic GMP, I mean, for example, the drug nitroglycerin, nitroglycerin. So what is this one for? Anyone knows this drug for? What is the function of this drug? For what problem? Heart attack, yes, heart attack. So cyclic GMP, as I mentioned earlier in the morning, cyclic GMP activity very fast. The action is very fast, much faster than cyclic AMP, in general speaking. Okay, and also interesting, we call the acetaminophen, or we call it Viagra. So this drug actually initially was developed for the cardio, but turned out also have the function for males problem. Yeah, so this is very fast action. Because the heart attack, the drug function cannot be slow, cannot be hours. It's usually come with a second or minutes, right? Nitroglycerin. So the cyclic GMP is through this pathway. It's very rapid pathway. Or even the phototransduction. Yeah, so our eye respond to the respond to the light. It's very rapidly. We need to respond very fast. So it's also through the cyclic GMP, but not cyclic AMP. Okay, so that's the same signal transduction pathway, but in terms of speed-wise, for example, they are different. So it's also involved in cell growth regulation and division. So in terms of speaking, classically, we call the cyclic AMP and the cyclic GMP regulation is like a seesaw. One go up, one go down, one go down, one go up, like a seesaw. But in detailed study, they're actually some sort of different. Yeah, they are not really counteract, okay? They are not really counteract. So the seesaw theory is like up and down, like a counteract, but it's not the case, okay? So cyclic GMP is much faster. Cyclic AMP much slower than like a phototransduction or the heart attack, all this mediated by cyclic GMP. And then one thing important is the cyclic GMP can be regulated by nitric oxide. Free radical. It is free radical, nitric oxide. Okay, so this so nitric oxide can actually activate the guanidine cyclase, or the cyclic GMP can be converted by another pathway. So the cyclic GMP can be activated through the two different pathways and then function as second messenger. Yeah, so the nitric oxide is really one of the very important free radical involved in signal transduction pathway. So that's why we say earlier is a crosstalk. The nitric oxide, like a gas, free radical, can through the signal transduction, like here, is a cyclic GMP, okay? And not only that, it can also regulate other pathway we will discuss later on. Okay, so this is a brief introduction of how the cyclic GMP regulated. So this is one of the example about the cyclic GMP, how the cyclic GMP. So the PKG actually in the rest time status is actually phosphorylated in the resting status. But when the cyclic GMP activator coming bound to the receptor, then the quickly dephosphorylation, rapid dephosphorylation, then transduce the signal pathway. So because it's already fully phosphorylated at the resting status, therefore when the ligand binds, it could quickly respond. So for the cyclic AMP is only until the ligand bind, then they start phosphorylation, start to transduce the signal. But for the cyclic GMP, is the receptor already phosphorylated, waiting for the activator coming, waiting for the ligand. So then the response much faster, okay, much faster. So these are very different. Yeah, so the target in PKG in the therapy, for example, the cardiovascular disease, the diarrhea is also the cancer, even the malaria is also the PKG pathway. Okay. So that's what I mean is try to give you. I think they have recording. I don't know if this is available to you guys. You can check with Dr. Kumar whether available. So that's as I said, every slide have a detailed description. Okay, you can have it as a reference. That's what I try to do. And talking about the cyclic CMP, cyclic CMP is actually also involved in different cellular process, can regulate the cell growth, cell division, the cell motility, gene expression, or ion transporter. So essentially, it's a lot of pathway regulation similar to the cyclic GMP, cyclic AMP. Yeah, it's similar to cyclic AMP, I'll put this way. So this is, yeah, it's also the same through activate the G protein couple receptor. So that's what we know so far about the cyclic CMP. Then the cyclic UMP, actually, it's okay. Cyclic UMP, we do not know so much, cyclic UMP. They can be in bacteria. That's what we know more. Is yeah, the first, as stated in the first line, the law is much less known. We don't know much. So it can be involved in the defense against infection by the bacteria virus. So that is phage. So bacteria prokaryote cell use cyclic UMP and a cyclic CMP as a defense system. So the signal come when the bacterial phage come in, they will activate this pathway, signal pathway for their defense system. So these are give you the reference, and you can take a look. You have interest in. Okay, so much about the serine phosphorylation, and we will switch the gear to the tyrosine phosphorylation. So the first tyrosine so as I said earlier, tyrosine phosphorylation actually was studied by German scientists during the World War II. We did not know, we never figured out what they are doing at the time. But the real study in the Freedom War, we put this way, after World War II is 1980. It discovered by essentially summer of 1979. They found the polyoma virus, the middle T and the VSAC are kinases. Okay, they have kinase activity. Then later on they find actually especially VSAC is a tyrosine phosphorylation, involved in tyrosine phosphorylation. This actually was determined by Tony Hunter. Then how we know it's the tyrosine phosphorylation? Because classically we know serine and cerulean. So then Dr. Hunter, he used a 2D paper chromatography. Used a two different buffer, pH 1.9. He initially used pH 1.9. So pH 1.9 could separate phosphoserine, phosphocerulean, and the phosphotyrosine. They run in the pH 1.9 differently. Okay, different. Then we modified into 2 dimensional from pH 1.9 plus pH 3.5. The first dimension we run pH 1.9. So 1, 2, 3, serine, threonine, tyrosine. And on the second dimension, actually the phosphotyrosine could separate much better from phosphotyrosine. So that we can really determine it. Then it was initially by Dr. Hunter, and they say, yeah, the middle T, polyoma virus, middle T, and the VSAC. So the VSAC has a counterpart in our body. What is that? VSAC is virus sac. Okay, so how about in our body? We call the CSAC. And it's very important kinase in the red blood cell. It's involved in the red blood cell activity formation. So the only difference between CSAC and the VSAC is the C terminal. So VSAC has a C terminal, can be fatty acid can be associated, then they can attach to the membrane. But the CSAC cleavage out that fragment. So CSAC cannot associate with the plasma membrane, so cannot transform the cell, but VSAC could. Okay, so that's what the theory by Dr. Michael Bishop and Howard Morris in San Francisco. They proposed cellular oncogene because of the VSAC and the CSAC. So they proposed cellular oncogene. That means the cancer gene, we call the cellular oncogene, is actually present in our body. In our body have oncogene already. So when time comes, it can activate, then we get the cancer. So it's a C oncogene versus CSAC versus VSAC. So both of them been awarded Nobel Prize. I don't forget which year. I don't remember. Okay. So the tyrosine phosphorylation is actually they are actually associated with the receptor tyrosine kinase, or some of them is non-receptor tyrosine kinase. Okay. So like the CSAC. That's what I'm okay. Yeah, this is the CSAC. Talking about the CSAC. Okay. So this is the receptor one. Then the receptor tyrosine kinase, like epidermal growth factor receptor or the PDGF receptor. Okay, parental-derived growth factor receptor. They're all tyrosine kinase, or even the insulin. Okay, insulin receptor. Yeah. So this is the function of the protein tyrosine phosphorylation. There are so many going on. They can, yeah, like the EGF, PDGF, FGF, they can involve the cell adhesion, cell differentiation, cell cycle regulation, gene expression. So the function is so detailed. There are so many functions they can be involved. Then insulin, here I use a different color here. Insulin is receptor is a very thick protein kinase, insulin receptor. So initially it was reported insulin receptor as a serine threonine kinase. But turned out insulin receptor could function as a serine threonine and tyrosine kinases. They can phosphorylate everything. Depend on the condition, depend on the growth condition. Okay. So initially we thought that insulin may be involved in regulation in the glucose metabolism, right? So the type 2, type 1 is really insulin issue. But now we actually don't know insulin should be classified as a hormone or a growth factor. Because when you add to the cell culture, the cell growth increase. So it fits the definition growth factor. But in addition to growth factor, insulin receptor can be involved in glucose metabolism. So it's a metabolic regulation. Okay, so it's a endocrinology study or it's a growth factor study. It's okay. We don't need to worry about that. We just try to find out its function. Okay. Yeah, so this is some sort of a brief comparison of tyrosine phosphorylation and serine cerulean. So this is tyrosine phosphorylation of the transmembrane protein. The serine cerulean usually are cytosolic protein, but tyrosine phosphorylation kinase also have so cytosolic protein like a SAC protein. We just mentioned SAC protein also is a cytosolic one. Yeah, so in here just mention says the cytosolic protein tyrosine phosphorylase is SAC protein. Then the receptor is the EGFR or insulin R. Then the serine cerulean could be the TGF beta or the PK, PKC. Those are the cytosolic. So roughly, you can say tyrosine phosphorylation, most of them are the transmembrane protein, but serine cerulean are the cytosolic protein. Okay, most of them. So this tyrosine phosphorylation, as I mentioned earlier, is they all through the protein-protein interaction, through the SH2 domain binding, SH3 domain binding, yeah, so the PTB domain, SHPH domain, PTB domain, SH2 domain. So they all through different domain of the interaction. Okay, they don't through the ion, it's a domain interaction. So this is a tyrosine phosphorylation also through the G protein activation. So we call the G protein coupling receptor CPGR. It's a cyclic AMP dependent, right? And involved in serine cerulean. But the tyrosine phosphorylation also involved in the G protein, GDP, all require the GDP. This is ROS oncogene. Okay, the ROS oncogene ROS oncogene been activated by the GDP. So classically, we call this is a small G protein. Then the GPCR that require the GDP, we call the large G protein because that molecule is bigger and this is small. The ROS oncogene only has about 20 kilodalton. Then the large G protein actually much bigger. I don't even remember, at least 70 or 100 kilodalton, that range. So that we call the large G protein, small G protein, and both of them require the GDP. And actually, ROS oncogene is the first human oncogene being identified. Okay, being identified. So it's a large G oncogene. So even they use a GDP activation, but the downstream phosphorylation through use ATP. Yeah. So the ROS, yeah, here say by Dr. Cooper and Dr. Weinberg, they actually discover three group actually discover the ROS. They have the HROS, NROS, KROS. Yeah. Okay. So Dr. Weinberg is actually put down here. Dr. Weinberg is actually the only scientist discover oncogene and tumor suppressor. He actually discovered the ROS oncogene and also the EGF new oncogene, HER2 new, is also cloned by Dr. Weinberg's laboratory. But he never received his Nobel Prize, unfortunately. People say, yeah, he's the one study, and then why he did not get it? Yeah, so sometimes it depends on how lucky you are, okay? But I always told my students, when you try to make the discovery, don't be the first one. Don't be. Why? Because the first one, when you make the new discovery, very difficult to publish a paper. People always challenge, is that true? So the reviewer keep challenge. Then you can keep revising your manuscript. But usually the better one is the second one. Okay? Or the third one. Don't be the fourth one. Why? Don't be the first one. It got to be the top three. Make the new discovery. Anyone knows? Nobel Prize only award to 3 maximum. Never award to 4. Okay? So usually 2 or 3. But the first one very difficult to publish, so the second one is the easier one to publish because you can cite the reference. You can cite the reference and say, yeah, people publish already. Then the third one is still okay, so you are still on the list eligible for Nobel Prize. But the first one, no, unfortunately, sorry, you are not eligible. Okay, so try to make a new discovery ASAP. So this is what give you some of the idea. Yeah, Bob Weinberg is really identified the ROS oncogene clonate, and then also the new, the HER2 new, ERB2, EGF, HER2 new. So 3 groups identified at the same time. So Bob Weinberg actually identified from the virus called the new, but they are the same protein. But he never got the Nobel Prize. He was so depressed. So this is a signal pathway from ROS to the MAPK. Then, yeah, so it's also going to the nucleus, regulatory protein, not only in the cytosol protein. So we showed the figure earlier. pKa go to the nucleus. Protein kinase also go to nucleus. And the much striking to us is we still don't know how the ERB2, for example, or the EGFR on the plasma membrane, the molecular weight is so big, 170 to 180 kilodalton. They can be found in the nucleus. They can be discovered, identified in the nucleus. How? How a protein from plasma membrane with such a huge molecular weight, 170, the higher than 150 kilodalton, transport into the nucleus? We still don't know the mechanism. Okay, we still don't know exactly. Yeah. We don't know exactly what they are doing. Yes, they do have a signal, but the point is how they transport, how they translocate it, get into the nucleus. Okay. Yeah, so this insulin receptor, that's what I mentioned it earlier. It can be serine threonine phosphorylation, can also be tyrosine phosphorylation. They can be involved in metabolism, like glucose metabolism. They can involve in the cell growth. So we add the insulin to cell culture, cell growth increase. So it's growth in regulation. And then definitely without the insulin or no insulin function, then we have the glucose metabolism issue. We got the type 1 or type 2 diabetes, right? So one of the mystery, we know that insulin receptor for so many years, I think since 1960s, but we are still working on it to try to find out exactly what is the function of the insulin receptor. Yeah. So then, not only that, you can see the crosstalk, the serine threonine phosphorylation, the tyrosine phosphorylation. Not only they have a crosstalk, for example, the G protein can come to the MAPK kinase pathway, then the downstream here, the calcium. So all this actually, as if we are going to just try to understand one pathway, we cannot figure out the entire picture. So only until we understand how the entire crosstalk, then we may understand the better on that picture. And not only that is because we understand the pathway, how they regulate, interact. Therefore, we can identify the potential side effect by a new compound, a new inhibitor, for example. We have a lot of compounds. We can inhibit the receptor or something, right? But why we cannot use? Because adverse effect, the side effect too much. So then we cannot use it. One is could be the poison, the toxicity issue. One is the adverse effect. Too much side effect, then it's not worthwhile to give to the patient. So if we can cut down the pathway, then that means the compound more specific, then it's no problem. So for example, allergy. I don't know whether in India you have the allergy issue. In US, for example, we have the spring the seed, okay, the grass seed in the fall, we have the dust allergy. So for example, I do have allergy. Then the traditional anti-allergy drug, antihistamine, actually have a lot of side effects. When you have it, you feel sleepy, you feel nausea, you cannot drive. But now the new generation of antihistamine, actually, you don't see those kind of side effects. So what's the difference between the two? Because the new generation has less side effects. How? Because they modify the side chain. They become more charged. So therefore, the charged compound cannot get into the brain. They only function localized. They don't go to the brain. You don't feel nausea. Then the early generation, the drug can go to everywhere can through the cell membrane, even the brain cell. So of course you feel nausea, you feel sleepy. Okay, so these are the basic fundamental. We understand those basic fundamental mechanisms, then we can improve it. We actually can help out to improve the quality of our life much better. So I do carry, say, the antihistamine, the new generation one with me all the time, because I'm sensitive to the dust in the environment. Okay, I even sensitive to, say, the grass seed. When the fall, the wind blow all the dust, I get allergy. Then the new generation one, I can have it anytime. I don't feel sleepy. I don't feel nausea. I have no problem. I can still drive. So just the charge difference. Okay, so we understand the mechanism. That's how we can target to the mechanism. We can improve our therapy. We can improve our therapy. Okay. Yeah, so this is the insulin receptor. They can have the certain role functions, phosphorylation go into the glucose metabolism. The tyrosine phosphorylation go to the cell growth regulation. So this is a very complicated molecule. They have a lot of phosphorylation going on. Yeah, it's really a major issue on this protein. So the other cell, the kinase, is phosphatase. So I mentioned that in the brain, there are 2% of the brain protein are the protein phosphatase, calcinuric. Then we always think kinase activates cell growth. The phosphatase inhibits cell growth. But the phosphatase can also be the growth regulator. Like here, the slide shows it's by Nick Tons. Okay, so the phosphate involved in the T cell, the T cell activate the protein tyrosine kinase in the B cell receptor. In here, he gives an example in the B cell receptor. The B cell, we know what it's doing. What is the function of B cell? What is the function of B cell? Yeah, immune response, antibody, right? B cell. How about other cell? T cell. So where's the T cell? Yeah, no, where's the T cell? Which organ has the T cell? Thymus, yeah. And the B cell is in what? Bone marrow and what else? Spleen. Yes. So we use a mouse spleen to do the hybridoma monoclonal antibody. It's a mouse spleen. Okay. So in here, it's trying to show is actually the link kinase. All this the protein tyrosine phosphate is involved in this B cell development. So actually, the phosphate involved in the immune response. It's not downregulate, it's activate. Immune response requires phosphatase. So the phosphatase and the kinase, as I mentioned earlier, which one is important? They are equally important. They are not totally counteract. In some situations, yes, but not all the situations. Okay? They can be independent, they can do different. Yeah, so this is another example how the PTP1B involved in the growth regulation. Okay, so this actually, yeah, we mentioned a lot about in the tyrosine phosphorylation. We talk about a lot of Nick Tonks. He is the one cloned out the PTP1B. Okay, he studied the PTP1B when he was a postdoc. He purified the protein and then identified the PTP-B family, CD45. So the CD45 is a common antigen in what cell? In the immune cell. Yeah, the common is the CD45. It's actually the tyrosine phosphatase. Okay, so this is about the tyrosine phosphate involved in the immune response development. But then, okay, serine serine phosphatase also play equal importance in the immune response. Serine serine phosphatase, because the transplantation, we have organ transplant, right? So because if you transplant an organ from one person to another person, the recipient have immune response. We need to give the prescription to suppress it. So what is the prescription doing? Suppress protein phosphatase. Okay, so far the best suppressant for transplant is a compound inhibit the protein phosphatase. So essentially now we can see the protein phosphatase involved in immune response, involved in immunodevelopment. So it's not only a phosphatase, a protein to counter other kinase, but it has its independent function to regulate cell growth. Okay, so a lot of, we try to keep, I'm sorry to say, seems a lot of material I start to throw to you, even we have discussed very basic phenomena, very basic function, but I try to tell you what this pathway is doing, what this pathway is doing. Hopefully, the way I try to do is to stimulate your thinking. Very important. As for this morning, the Vice Chancellor say, it's really here, okay? When there was no instrument, how could they make all those discovery? How could they do it? Yeah, even in the biology, the same. Not only in the physics or in the mathematics, in the biology too. Why? They can do it. Yeah. Okay, so this is another example. Is we always thought, classically we thought phosphatase is less important than kinase. But it's actually, it's not a totally countdown. They have their function. Immune suppressor for transplant is actually a phosphatase. Here now we're talking about the B cell development. It's actually a phosphatase function. Okay, so yeah, so far it's really how the signal transduction going on now is the phosphohistidine. The phosphohistidine because it is a phosphate and nitro bond. So the phosphohistidine is heat and acid sensitive, very, very sensitive. So it's very difficult to study as the other one, okay? So currently we don't know much about it. We know the NME1 and ME2, mainly those two proteins, two kinases, histidine kinases. So it's in the fungus infection, they can be involved in really the bacteria infection, those kind of pathway. In eukaryote, it's much less study in eukaryote cell, but mainly in the prokaryote cell. So then the kinase, histidine kinase, the difference is two subunit. It's a kinase subunit and the responsive regulator. So the two put together, then it's really the histidine kinase. That's what we know so far. Okay. Yeah, so this is histidine phosphorylation can be, and the different, in the location here, the N or it could be this N1, N3, or here and then here, both of them. So they can be phosphorylated, managed in the nitrogen. Okay, so we are briefly went through it, really try to let you understand what is the function, what is the basic concept, and I try to throw in what the updated what we know. Now we are talking about the exosome that I mentioned earlier. We do have an exosome. It's a vehicle butting out from a cell. Then the size is bigger than what we understand, and it contains a lot of materials. Not only the protein, they can have RNA. So be honest with you, when I was asked to review a manuscript about the exosome, I was very confused. Not only I was confused, actually most of the reviewers were very confused. Is that really exist? Because this discovery only until 1983. 1983, okay. So it's a very confusing because it never happened. How can a cell bust out a small vehicle containing the protein or the RNA or DNA inside, and those can do the signal, can transfer the signal to the next cell? How they recognize it, how they identify their target protein. But as a reviewer, what is a reviewer function? You review a manuscript, you say, yeah, this manuscript will be accepted or not, right? What is the criteria you are doing? What is the criteria? How you review it? So as a reviewer, I review a manuscript is whether the result support the conclusion. Okay, very simple. Very objective criteria is whether the result presented support the conclusion. That's what they try to say. If yes, even you don't believe, say you don't believe the concept or very novel concept, you still need to accept it. This as a reviewer, serving as a reviewer. This is always my principle, my concept. So even I don't believe, I was confused. I sometimes did not believe it. But the data support their conclusion. I can only say, yes, it's acceptable, but revise some of the figures and not good quality or something, but I still need to accept it. Some reviewers just based on what they don't believe, then they reject the manuscript. Some. So for example, I mentioned to you about Tony Hunter. I know him, we know each other. Then I studied phosphatase, he studied the kinase, right? So he rejected my manuscript on phosphatase. So I call him, I say, hey, Tony, what's happening? Why my manuscript, the critique, I said, the critique has a problem. He said, personally, I don't believe it. Therefore, I reject it. We went through all the critique. I have 4 pages, 6 pages, 3 reviewers. Each reviewer give me 2 full pages, single space one. So I went through it. I said, yeah, they actually did not agree to one question. So that's what then interesting. He remember the copy of critique. He even used handwriting. Say, hey, Ming-Hong, call me when you have a question. You have time, we call. So I call him, of course. Then after all the discussion, he said, Ming-Hong, yeah, I think you are right. Your protein indeed is a phosphatase. Then I said, oh, okay. Then if I revise it, respond to the critique, then should my manuscript be accepted? He said, no. He said, because I don't believe it. I said, you are a scientist, right? He said, yeah. I said, we are scientists. We make the judgment call. We base on the data. We don't base on our believing. The believing, if we make the judgment call based on our believing, that's a religion. That's what I learned from my professor. That's a religion. It's not science. The science is based on the data. He said, no, I don't care. I'm a senior reviewer. I'm the corresponding editor. So I don't believe believe this protein could do this function. That's it. I reject it. He said, doesn't matter, you can answer it. So of course, we are not friends anymore. No, this I just try to explain to you what to do. But then another issue I can tell you, when I submit a manuscript to another journal in British journal, biochemical journal, it's very classical journal, very studied biochem journal. Yeah, the reviewer the same has concern. And I reply, at the time I was a junior assistant professor, so I answer it, I do all the explain what they request. So I send it back rebuttal. After 3 revisions, the editor-in-chief sent me a letter say, even I don't believe it, but you answer all the reviewers' critique, and your data support your conclusion. So even I don't believe it, I accept it. Okay, so this is the fundamental as a scientist. He said, yeah, personally, I don't believe it. But since you addressed all the concern, all the questions to the reviewer, personally, I don't believe it, but accept it. What? Your question? Yeah, question? Yeah, no, it's just I try to explain to you. It's not say I try to tell you what's going on. It's a personal, it's as a scientist, how we should behave. Because you are a graduate student, right? Most of you are graduate students. Then you continue your research. In the future, there's a very high possibility you are going to deal with all this, serving as a reviewer, consultant for journal, whatsoever. You will continue, later on become as a professor, right? So you are reviewing a lot. So the key is you got to have your own principle. What is your own principle? The subjective principle is the data support the conclusion. That is a reviewer's judgment call. But not say, I don't believe it. Or personally, I don't believe it. I say, yeah, but why you don't believe it? He said, I just don't believe it. So that's what I explained to him. I said, Tony, even he's senior to me. I say, you are a religion person or you are a scientist. Okay. So that's what, so even when I deal with exosome, I don't study it. I was asked to serve as a reviewer. The data support what he tried to propose. Then I can only accept it. Of course, some of the data I like them to revise it to a better quality. But beyond that, I accept it. Yeah, even now, be honest with you, I still have a lot of questions about the exosome. Or even now. But there's a lot of study going on from good lab, very solid data. And even personally, I do have some question, but I still need to accept the fact it is there. Just I still don't know what it is, that's all. Okay, I need to say I still don't know what it is. As a scientist, we don't know everything. We don't know everything. We just know what we are doing. We may not know everything, but we need to honestly accept the concept. We don't know everything. Yeah. So this is the exosome. They are the same, has a lot of receptor. They're containing including the protein. Yeah, they have the protein. They have the RNA, the microRNA. So they can up to, a single exosome can have about 20,000 molecules inside. This is incredible. So how can they function properly? So this is really, so the same is that they need to find a receptor and then bound to the receptor and then release the material to the cell for the function. Okay, so it's not only in the cancer cell. This actually initially found out in the red blood cell. So it's a normal function too. Yeah, so exosome involved in the normal cell function. So this is a detail how the process from A, B, C, D, E, how the exosome get into the cell to release the content for the function. Yeah, so how they can come out with the RNA and the protein in the vehicle. And then they can go to the next cell and have membrane fusion and budding in. release mRNA, release RNA doing the function. It's still a lot of going on. To me, as I say, I'm not in the field, so but it's just they are there. Just we need to do more to understand more. Okay, so this is what is an exosome hypothesis and what their description, their function, they are going on. So they even mentioned they can have a crosstalk between the embryo and the maternal and the mother. Yeah. So they serve as a messenger between the baby and the mother. So this is another scheme talking how the cancer cell, they actually release the vehicle, the exosome, into the recipient cell and then can transform the recipient cell become a cancer cell. So yeah, this is all the function they can do. So far we know about it. Okay. Yeah, how could a small vehicle in the cancer cell we can understand it's much better, but for a normal cell, how could that do it? That is a challenge. Okay, so the regulation. So we're talking about the regulation, the signal cascades regulation. They can have a transcriptional regulation. They can have post-translational modification. They can have catalytic activation or inhibition. So all this, so it's the different level of the regulation. They can have gene expression. So this is what regulate, yeah, in the different level. So it's similar to the regular protein. There can be the gene turn on, the post-translational regulation, could be the phosphorylation, restoration, or could be the glycosylation. Okay, and they can be activated, could be oxidation reduction, could be binding partner interaction, or they can be whatever. Then similarly, they can be degraded. So the signal transduction pathway not only say we see the signal going in, but then how they are interact within inside the cell. So this is actually the whole one chart, one figure could give you all the story about how they function. And he used this chart actually used a sample. It's a dual-specific protein phosphatase. Okay, so the phosphatase can dephosphorate tyrosine alone or serine and tyrosine. So this is an example of dual-specific tyrosine phosphatase. Yeah, so they can be regular at a different level, either at the gene transcriptional level or the protein phosphorylation, everything. I think that's what I have for today, okay? So I know it's a lot of basic material. Some of you have it already, but yeah, some of you have some of the background, but hopefully they could refresh your memory and then they have some idea. The signal transduction pathway is really a crosstalk from here. I mean, a different, yeah. It's not only a one-way signal. We got to understand, okay? It's not only one way. Not in the freeway, it's a one-way signal. They can go to every direction. They can come back, they can go forward. And this is the biggest protein phosphorylation. And in today's talk, we already discussed some of the crosstalk related to the gene regulation. For example, serine phosphorylation or tyrosine phosphorylation, they can activate the gene expression. They can go to nucleus, turn on or turn off gene expression. Okay, not only for classical, we know just only the protein phosphorylation. No, it actually can go to turn on the gene. And how the growth factor receptor, such a huge protein with a 170 kilodalton protein, it's very difficult to translocate it and then into the nucleus. But indeed, they are there. And not only one group discovered it, there are several groups. I have several publications. But we don't know how the regulatory mechanism which receptor can transport into the nucleus. We don't know. And for insulin receptor, for example, it's a serine starch and serine phosphorylation, and also tyrosine phosphorylation. But when it's only for serine, when it is only it's for tyrosine, how to fine tune, go for this side or go for the other side, it's still have a lot going on too. As I say, I can only say we still don't know much when it will go there, when it goes to this way, and when both will be turned on. Okay. And tyrosine phosphorylation, we're talking about the kinase, we're talking about the phosphatase. Yes, in general speaking, phosphatase counteract kinase, even serine the same. They have much complicated protein phosphatase system. Yeah, they have protein 1, phosphoprotein 1, phosphoprotein 2. They have a lot of membranes. They have catalytic subunit, they have regulatory subunit. They have a lot of regulation going on. And the tyrosine phosphorylation, in comparison, it's a much simple study. It's a much simple regulation. But still, tyrosine phosphatase not only counteract tyrosine kinase, but also has their own independent function involving immune response, immunodevelopment, B cell development. So we don't need to, I don't like you to think, yeah, phosphatase only dephosphorylation, that's it, okay? It's actually involved in a lot. As I just mentioned, the best prescription drug to suppress immune transplant rejection is an inhibitor to protein phosphatase. Then in our brain cell, 2% is carcinogen, protein phosphatase, and regulate our memory. It's not a kinase regulate, it's a phosphatase regulate our memory. Okay, so at least for today, I'd like you to have a concept. Kinase phosphatase, yes, they do have the counteract, but they do have that independent pathway. They can have on their own identity, not only interact. And they do interact, but when they interact, when they have independent identity, it's another level of regulation. I think we need to understand. Okay? Okay, so far, any question? Yeah?
The immune part of phosphatase. So could we say that phosphatase has a role in immune evasion in cancer? Like we know that cancer cells, they evade the immune system of the body.
Yes.
So could we say that there could be a role of phosphatase in that immune evasion process?
No, cancer cell is some sort of slightly different. Yes, you are talking this maybe whether they have a. It's a very good question. I don't think anyone really get into it. Okay, I can clearly I don't remember anyone really get into it to see whether say the protein tyrosine phosphatase some sort of expression being suppressed because of the cancer cell. But yeah, currently we know the cancer cell could survive through the immune response because they have we call the immunosurveillance system, immune surveillance, because the cancer cell has more salicylic acid. So salicylic acid, you know what is the temperature? What is the charge? Not temperature. It's a negative. So it's highly negative charge. So it's some sort of cover the cancer cell. Then the immune cell is some difficult to recognize it. It's highly charged. So that's what we call the immunosurveillance. So it's classically been proposed for a long time, but recently, yes, indeed, that is the case. Yeah, so salicylic acid is one of the important ones. For example, we did do the experiment. The cancer-associated antigen is actually, so on other side of the coin, we have now a lot of biomarker we call the cancer-associated antigen. We see that increase, then we know this patient might have cancer. Okay, we call the cancer biomarker. So a lot of cancer biomarker, they can activate it in the patient circulation because highly glycosylated by salivation. So if we use the enzyme neuraminidase to remove the salicylic acid, actually those biomarkers, boom, quickly disappear from circulation. We no longer see the biomarker. Yeah. So the glycosylation is another regulation. It's even more complicated than signal pathway. Signal pathway, we know the cyclic AMP, we know the cyclic GMP, one or the one. By glycosylation, even one of my colleagues, he's a really glycosylation protein, glycoprotein biologist. I never really figure out all those core 1, core 2 enzyme. Yeah, it's more complicated and it seems not so systematically regulated in a way. I will put it this way: is they are not so tightly regulated as we know the signal transferring pathway. Yeah, it's more loosely controlled, regulated. Okay, but if you have a very good question, I don't think anyone really figure out whether the phosphatase are involved because, as I said, there's more people study kinase. It's much easier. There are many less people, very few people in phosphatase field. Yeah, very few people. So the study much more difficult. So I think no, it's actually this. Yeah, we need to think about that possibility too. Yeah, no, it's a good question. Any other question? Anything else? Anything I can answer beyond the science? Okay. So I think I did explain to, earlier before the class, I did talking about how to go to abroad to study. Yeah. So any question, any further question on that subject, I will be happy when I'm here. We can chat about that too. No problem. Okay. I will try my best to answer to you since I can compare to you the difference between the US system versus Europe system. So I know a lot of students go to Europe, England, or British, okay, go to Canada or US. So they totally different system, or even Japan. Yeah, they are very different system. So luckily I know them pretty well because I have a lot of colleagues there from different area, different regions. So that I'm the one like to talk. So even I was complained by one of my former chairmen say, Ming-Fong, why our faculty member talk with you more than talk with me? So I know I had a big problem. I said, yeah, I like to talk, that's all. Yeah, so as I say again, the science, the answer coming out from your own mouth. You have a question, you ask, you open your mouth, ask, you got the answer. But you don't ask, you never got a real answer. So don't feel shy, okay? As I said, I don't have all the answer, but I can give you some idea what the direction could be. I don't know all the answers. The science, I said myself, I don't know exosome, what it is. Even one of my colleagues, his office next to mine, he's an exosome expert. I just told him, I say, he actually was from your country too. I say, yeah, your exosome confused me, okay? You should do more to convince me, say, it is there. But when I review my script, yes, I did accept it because I base on the data. I don't base on my belief. I don't base on my belief because I had trouble. I was treated by that way, so I don't do the same thing to other scientists. Yeah. Okay, now I think we can dismiss the class for today. So tomorrow we have what? 10:30? Yeah. Let me check the schedule. Oh yeah, 10:15. 10:15. Okay, 10:15. Yeah, tomorrow I'm going to talk about the redox. I did not know, I did not believe either, but now I'm a total believer. I did not believe the free radical. And now even one of my colleagues, he said, I still did not believe the free radical. I say, yeah. I said, but I said, when I was a time with you, I did not believe it, but now I totally believe it. It's really, when you get into it, you dip into it, you understand it, then that you accept the concept. Yeah, and one of my professor told me, say, I should not use belief, because the belief is a religion. It's, we propose, we accept the hypothesis. It's not use the word belief. I somehow concur with him. Okay, I somehow concur with him. But you know, sometimes how you are going to distinguish the two, I don't know either. As I say, I told you all the time, I don't know. In reality, yes, I don't know. Okay, thank you. Yeah, thank you. Okay, so this is your, oh, so the slide you will save it. Yeah.